And we are now on YouTube. So we switch off all our cameras and microphone except Eva and Olivier.
Okay, bye-bye.
I have nothing to do, right?
No, you just stay there. And I will start in 1 minute.
Yeah.
Okay, so I propose to start. Welcome to everybody to the second day of the ICMNS conference. I'm Eva Löcherbach. I'm speaking to you from the surroundings of Paris. I'm working in Paris 1 University. First word, today's program is a little bit longer than yesterday because after the mini symposia session, there will be a session on the future of ICMNS, to which you're all welcome. It will start at 10 minutes past 6 o'clock Paris time. And if you have any suggestions that you want to submit, there is a form on the webpage that you can still use for doing that. Okay, so that will be at 6 o'clock. We will start now with the second plenary talk, and we are particularly happy to introduce Olivier Fujavas as second main speaker. As you all know, he has initiated this series of ICMNS conferences in 2015 in Saint-Jouan-les-Pins. Olivier is a member of the French Academy of Science. He is a research director emeritus at INRIA in Sofia Antipolis, where during a long time he was the director of a research group called Neuromatcom, and that's a project that he actually initiated. He is, I would say, one of the leading figures of the mathematical neuroscience community, and actually he did a lot for this community. For example, with the creation of the two journals, Journal of Mathematical Neuroscience, and then the follow-up, the Mathematical Neuroscience and Applications. With his main scientific contributions, Olivier has built a bridge between the deterministic world, so the world using dynamical systems theory, bifurcation analysis, and the stochastic one, using stochastic processes mainly by the means of large deviations. He is interested in a lot of things, among others in the asymptotic properties of large systems of interacting neurons, the rigorous proofs of the mean field limits, in the theory of neural fields and the applications to the modeling of visual perceptions. He's also interested in plasticity, in learning. He also worked on random synaptic weights, Hopf field neurons, and so many other things that I'm not going to talk about all these. Olivier received for his scientific work several awards and prizes, in particular the Asriel Rosenfeld Award for his work in computer vision. And today he will speak about neural fields, from vision to consciousness, a prospective mathematical perspective. And so we are very curious about what you're going to tell us, Olivier.
Thank you very much, Eva, for this kind introduction. And thank you to all of the committee for inviting me to this conference. So you said the title, I won't repeat it. This is an outline of my talk. I'll start with a short introduction. Then I'll discuss the mathematical study of the neural field equations. Then I'll see some applications, including symmetries to visual orientation. I hope to have the time to talk about stochastic neural fields And then I'll dive into the question of consciousness. I haven't worked on consciousness, but I think it's something some people in our community should work on, and I will conclude. So the introduction, mathematical neuroscience is really, the goal is really to develop predictive and possibly falsifiable, a la Karl Popper, this British philosopher, theories. One of the most famous ones, one of the most famous theories, such theories, is found in the work of Hugh Wilson and Jack Cowan, published in the years 72 and 73. Here are pictures of Hugh and Jack and their papers, the landmark papers that they published in Biophysical Journal in 1972 on neural masses and on neural fields in 1973. Let me flash the celebrated Wilson and Cowan equations at you. They are written here on the slide. If E of t and I of t are the average membrane potential of excitatory and inhibitory cells at time t, then you have those two nonlinear differential equations. I just comment a little bit. The first one, tau d dt equals minus e. That's just an exponential decay toward the initial condition. And then you have a linear combination with weights j sub e, j sub I, e of nonlinear functions of e and I. Se and Si are the usual sigmoids. We'll hear a lot about sigmoids later in the talk. And IEX is the external input to the excitatory neural population. So this is the voltage-based neural mass model. There are also activity-based neural mass models. So this is what a neural mass is, and we'll see neural fields a little bit later. So after this very short, brief introduction, I will talk about the mathematical study of the neural field equations. It starts with the work of Wilson and Cowan again in 1973 and followed by Amari, a Japanese researcher, in 1977. And it was followed by many attempts by theoreticians and mathematicians who tried to characterize the solutions. There's a long list here, Breslov, Coombs, Amontrot, and many others, which I won't have time to read. Today, I think it's accepted in the mathematical neuroscience community that the correct framework for analyzing these equations is functional analysis, because you can use such functional spaces as Banach, Hilbert, Sobolev, which are quite handy. You can use fixed point theorems to prove existence and uniqueness of solutions. You can use spectral theory to analyze stability. You can use bifurcation theory in order to analyze how the solution changes when you change parameters. Let me write them for you, show them to you. They resemble the previous equation, the Wilson and Cowan equations, with some change in notations. So I'll read it. V dot of x and t equals minus v of x and t plus j of t applied to s of lambda v at x plus some external current. And there is an initial condition v at time 0 is equal to v naught. So what are these quantities? V and v naught are p-dimensional vector functions, which are defined on a set omega, which can be an open, bounded, or unbounded piece of R2 or R3, or a more complicated space. And they represent average membrane potentials of the neural field, like in the Wilson-Cowan. They both belong to some functional space F. Now, J of t, this is the operator notation I have here. J of t is a linear operator which maps the functional space F to itself. It's also called the connectivity kernel. And it describes the intensity of the projections from the masses at x prime to the masses at x. And this is the linear superposition integral, which is shown at the bottom of the slide. Now, let me comment a little bit about the other quantities that appear in the equation. S is a sigmoidal function, which is basically regular and bounded, like in the Wilson and Cowan equations. Lambda is determines the stiffness of the sigmoid, an important parameter. I will use it as a bifurcation parameter later. And X, which lives in omega, represents space or features that are more complicated things. Now we have to decide on F. So there are three criteria that we can follow for choosing F. The problem should be well posed. The equations should be solvable. We should also worry about the biological relevance of having membrane potentials functions living in F, and it should also allow numerical computations. So one of the usual choices is the Hilbert space F equals to the set of square integrable functions values in RP and defined on omega. You can also, if you want, impose more spatial regularity, but I won't need it in this talk. J, the connectivity kernel, also lives in an L2 square, a set of square integrable functions. And if it lives in that set of square integrable functions, then J of t defines a continuous linear operator from F to F. So the superposition integral is well defined as a mapping from F to F. Now I rewrite the neural field equations as an ODE, ordinary differential equation, which is defined on the functional space F as dv dt equals minus v plus r of tv, and initial condition is just a function v naught in F. And r of tv is the sum of the operator j of t applied to s of lambda v plus the external current. And in a 2008 paper, SciEM paper, together with François Greenberger and Jean-Jacques Slotine, we proved that if J is a nice mapping, continuous mapping from R plus the time to L square, and its F norm is bounded for all T's, and if Ix, the external current, is also a smooth continuous mapping from time to functional space, then for all initial conditions, there is a unique solution V defined on R plus for all time and continuously differentiable of the Cauchy problem. Sorry. I would like to mention briefly here some closely related work by Roland Pothast and Peter Baumgraben, published in 2009 in the Mathematical Methods in the Applied Sciences. Now, we have existence and uniqueness in this case. What about boundness and stability? Well, we also had this results with François Greenberger and Romain Vels in neural computation. If the external current is bounded in F norm, then the solution is also bounded. Your membrane potential does not explode, doesn't blow up. And furthermore, if the product of the stiffness by the spectral radius of the symmetric part of the operator J is less than 1, then every solution is globally asymptotically stable, and in particular, it is unique. Okay, so we would like to learn a little bit more about these solutions. And in particular, I would like to state a few results about persistent states. They are important because persistent states or stationary solutions that do not depend upon time are pretty good models of memory. So I will have to assume that the external current and the connectivity function do not depend upon time. And I will rewrite the neural field equation, no more time derivative, as f of v and lambda. Lambda, remember, is the stiffness of the sigmoid. So two main questions we tried to answer in the past. How many solutions can there be? We saw that under some conditions there was only one. And what is the manifold of solutions when you vary the parameters? So we have this important proposition in a paper with Romain Vels in SciEM in 2010. If I define the set of solutions for a given value of lambda as such here, b lambda as the set of solutions, which I note vf lambda, then first of all, we proved that the norm of the difference with the solutions for lambda equals 0, so it's a flat solution, is bounded by a function, upper bounded by a function of lambda, which is also bounded, which means that the solutions live in the white part of the right-hand side picture, which shows the horizontal axis is lambda, and the vertical axis is this infinite dimensional space where V lives. And of course, we just represent the norm of V. So they are bounded in that space. And the second thing we proved is that if the number of solutions is finite, then this number must be odd, as shown in the picture, 1, 2, 3. And we proved this by using the Luray-Schauder degree theory. The third result we had was that if you consider two values A and B of lambda, shown on the right-hand side picture, and if I consider the sets B lambda times lambda, so these are the vertical lines and points on these lines corresponding to the solutions, then there's always a connected component which intersects B sub A and B sub B. So the left-hand side of the picture is impossible. There's no path, but the right-hand side is what happens. The green path connects B sub AA to B sub BB. And we did that, we proved that using the Luray-Schauder theorem. So we have pretty good global information about how, about the geometry of the stationary solutions. That was in that paper. Okay, so that's the end of the mathematical study of the neural field equations. I want to apply this to visual orientation sensitivity, or how symmetries make things more complicated. So we know from neurophysiology that visual area V1, shown in red in the slide, is organized as a set of orientation sensitive columns and hypercolumns. This is the beautiful work of David Hubel and Thorsten Wiesel in 1974. And this is shown in the right-hand side, the right picture. Orientations, visual orientations in the retina are coded in color. So color is a visual orientation. And if I plug in an electrode here at this point on the cortex, this is a visual cortex of volume, this is the surface here, I notice that the neurons respond only to the red orientation. Okay, and if I go deep in the cortex, this sensitivity doesn't change. But if I move about, then this sensitivity will smoothly change, moving from red to purple to green. And along these white curves, it's pretty smooth. It's constant orientation. And you can see that these curves meet at some points which are singular points, and these points are pinholes, called pinholes, and will play a very important role in what follows. Okay. If I look at the cortex from above, I see this map of colors, and this is called the preferred orientation, or PO map. And it's periodic because of the pinwheels. This periodicity will turn out to be important. Now, the neural field model I can write for V1 is very simple. It's a classical one, as before. dV dt V equals minus V plus the superposition integral. I changed the lambda to sigma to be compatible with the figures I had at my disposal. And now we will focus on the connectivity function J, which I will assume to be the sum of two parts. A local part, J lock, which is a translation invariant. I forgot to say that X and Y are points on the surface of the cortex, so it's translation invariant, plus epsilon, where epsilon is a small number, positive number, much smaller than 1, a long-range connectivity function, J sub LR. Okay. So J lock, the local connectivity function, is a Mexican hat-like. So it has local excitation and local remote inhibition, plus and minus. Now, the bifurcation analysis of the homogeneous model, i.e. the model when epsilon equals 0, is very well understood. We solve in the square omega with periodic boundary conditions. Therefore, omega is actually a torus. And we know that there is the emergence of Turing patterns as shown on this bifurcation diagram. The horizontal axis, again, is the stiffness sigma of the sigmoid. The vertical axis is the infinite dimensional solution. And that is well known. For example, see the paper by Coombs, Baumgraben, and Pothast in the book they recently edited in 2014. We know that there's an emergence of Turing patterns. There are stripes, as shown here, and I have a problem with my mouse. Stripes and spots. So stripes are always unstable. Spots are stable on a very short interval, and then you have two subcritical secondary branches that recover stability. Stable is shown in orange or yellow if you on your screen maybe. Okay, so this was with the symmetries of the local orientation, local connectivity function. Now, I said before that because of the translation of invariance, the solution lives on the torus. Now, I will assume, I haven't talked about the PO map, but I will assume that it is periodic, so it's approximately periodic, and the period is the same as that of the periodic solutions. This is fairly well supported by biology, paper by Kenneth and colleagues. So what I'm showing here is the PO map. Again, remember that orientations are coded in color. The black lines surround the hypercolumns, where at the center of the black squares you have a pinhole, and the white lines correspond to the square, the fundamental domain. So the pinwheels are shown either in black or white. And notice that if I rotate, if I rotate about a pinwheel, the color will change in different ways. In other words, black pinwheels correspond to a clockwise changes in color, whereas the white one counterclockwise. So pinwheels are clockwise or counterclockwise, meaning that if I rotate about, then colors change differently. Now, let's talk about the long-range connections. The long-range connections will make things very interesting. J sub R is given by this interesting expression, which I will not comment accurately, but I will use the biology here. Again, we'll look at the PO map, and there's a center here, which is sensitive to purple color. So this is one orientation. Then through the injection of some dye, by tracing how the injected dye progresses, we can tell that these particular neural masses are connected mostly to other neural masses remotely, which are sensitive to pretty much the same orientation. Okay, so it's patchy and it's orientation sensitive. This is a diagram which is taken from the landmark paper by Breslov et al. In 2001, which shows that if I point X, I'm here, I have various columns sensitive to various orientations. And you see that this one at X is only connected to this one at Y with the same orientation. And the same here, this one has a different orientation sensitivity, and it's only connected to the one at Y, which have similar orientations. This is very, very important, and this is gathered in this formula. Now, when you study the bifurcations or the changes in the solution, and there is a group underlying your model, it's important to look at the symmetries of the PO map of your equations. So the most important symmetry here is the action of the rotations with center pinwheel on the PO map. So if I rotate a point X with center P of an angle phi naught of pi over 2, what is the value of the PO map at this new point? Well, it's very simple. You can check it by yourself, equal to the value of the PO map at X plus epsilon phi 0 by 2, that is epsilon pi over 4. And epsilon is 1 if P is counterclockwise. Remember that the pinwheels are counterclockwise or clockwise, and it's -1 otherwise. Okay, so if we want to study the changes in the solutions, we form a set omega with 2n+1 copies of omega naught. Remember the white square. The symmetries of that set are the symmetries of the square, D4. semi-direct product z modulo 2nz squared, since we are in two dimensions. So that's the symmetries without the PO map. And it turns out, very interestingly, that the PO map reduces the symmetry to one of the 17 wallpaper groups or crystallography groups, which have been studied by mathematicians since the 19th century. And further constraints from the PO map reduces this whole set of groups to just two, called PMM or CMM. And they are shown actually, they are shown here and here together with the PO map. Okay, so we have this extra group of symmetry that acts on the PO map. And what happens? Well, what happens is that we're going to lose some symmetries in the solutions when we turn the long-range connectivity on when epsilon is non-zero. So as shown in our paper, in 2015 paper in the JMN, by the way, the symmetry group gamma of the NF equation, neural field equation, is one of these crystallographic groups, P4M or P4, meaning what? Meaning that if I call f of v of lambda the neural field equation, and if I call little gamma an element of this group gamma, which is equal to P4M, then if I apply gamma to f of v and lambda, it's just the same. It's equal to f of gamma applied to v. So that means mathematically that the neural field equation is equivalent with respect to the group gamma. So it's well known when you turn on the long-range connections, the system undergoes symmetry breaking. The solutions will preserve only the symmetries of gamma or some subgroups of gamma. Okay, so we understand the symmetries. What about the dynamics? Well, the dynamics can also be studied. I will try to explain this briefly. Remember that the solutions live on a torus when there is no long-range connection. So I call T naught the unperturbed torus, which is V0 of X, and X is in omega. And we add, when you add the long-range connection, then you have a torus, flow invariant torus TLR, which will persist as long as the perturbations are small. Epsilon is very small with respect to 1, small enough. So this was shown, for example, in the beautiful book by Pascal Henson and Rainer Lauterbach, Methods in Equivariant Bifurcations and Dynamical Systems in 2000. So questions we can ask. So how many steady states do actually persist? And what are the observed phase portraits? Well, the topology of the torus is an important constraint. As shown here, we have this proposition in the same paper. If we assume that there is a finite number of equilibria on the perturbed torus, which are all hyperbolic, non-degenerate, non-zero real parts, and if epsilon is non-zero and the anisotropy is real, that is key, the parameter key, then there are at least 8 equilibria on the perturbed torus, 4 of which are saddles, and the other 4 are nodes or foci. And the proof is a simple application of the Poincaré-Hopf theorem that tells us that if n is the total number of equilibria, that is the sum of the number of foci and the sum of saddles, n minus 2s is equal to p, the characteristic of the torus, which is equal to 0, and hence f is equal to s. So then if we look at rotations, the rotations with respect to the pinwheels, we find that the number of foci is equal to 4. Therefore, we have 4 saddles. This is a very deep and very interesting theorem proposition. And actually, you can verify it numerically. You can draw phase diagrams like here, where you can, this is a simulation in omega. You have these 4 saddles here, and then you have 1/2 foci here, 1/2 foci here and here, that's 2 foci, and 1/4 of a foci at the 4 corners, that's another foci, 1 at the center, that's 4 foci and 4 saddles. Ooh. We cannot, so there's dynamics, I forgot to say, there's motion in here. Examples of cortical activations. These are simulations of the equilibria we superimpose the activity map, the membrane potentials, or S of the membrane potentials, to the PO map, and the PO map is semi-transparent. When the activity is very large, the PO map is transparent. The activity map is transparent, so you see the PO map, and you can represent this as a perception, if you wish, by averaging the PO map, with weighted by the activity. So here we see basically patches. Here also we see patches at different solutions, and here we see edges, only one orientation. So here we see all orientations, and here none. Okay, final remarks on this part. It's a pretty wide generalization of the early work of Armand and Cowan in Biological Cybernetics 79, and the landmark paper by Breslov et al. In 2001. And I would like also to stress the fact that, to me, group theory is really underutilized in neuroscience, and we should make more use of it. Okay. Taking noise into account, stochastic neural fields. Okay, so we know that neurons are intrinsically noisy because of ion channels, thermal noise, synaptic noise, and so on and so forth. So we would like to rewrite the deterministic noisy neural field equations a la stochastic differential equation, which I did here. dV equals the drift term plus the diffusion term. S and sigma are globally Lipschitz. S is bounded. Okay, and what is W of T and X? It's a stochastic process in two variables. T, it's time, and X belongs to Rn, which is our set omega here. So we have to define what do we mean by a solution, and it has to involve an object, this kind of integral, an object of that form. And I will try to explain this a little bit. Let me also mention that this approach has been used empirically, i.e. without worrying about being mathematically correct, by Breslov and Weber, Breslov and Wilkinson, Kilpatrick, and Ermantraut. So one of the approaches is to use the theory of Walsh stochastic integral, published in 1986, which allows you to cleanly define a white noise W dot on R plus times Rn, and then a white noise process, Wt of A, where A is a Borelian of Rn, and a set of functions, Pw, that can be integrated against the white noise process W, which is exactly what we need to make sense of this object here. Okay, so Walsh has done the work. There are some problems. Informally, you can think of the object W dot as a random distribution, very rough. So any solution of the stochastic neural field equation is distribution valued in the X direction, which is the space direction, and that's no good. So you can smooth that out by defining a slightly different noise, Gaussian random field, by basically convolving it with a function in L2. This is the integral here. And it's easy to verify that phi of x minus dot is in the set of integral functions against W. You can prove that W phi is regular in time, same as the Brownian path, i.e. Hölder continuous for alpha strictly less than 1/2. And Hölder continuity in space, which is also nice, can be enforced by imposing a very weak regularity in phi, which is indicated here. Tau sub z of phi is just translated by z. Phi is just a function. If you take the L2 norm of the difference, then it should be less than c phi, the Euclidean norm of z raised to the power alpha. So if you do that, if you have this condition C1, for all t, the function x goes to W phi of tx, it has an eta Hölder continuous modification for all eta less than alpha. That's fine, that's nice. Now we can rewrite using this W phi, we can rewrite correctly, as I will show, the stochastic neural field equation driven by spatially smooth space-time white noise, as shown here. And by a solution, we mean a real valued random field, as shown here. So you have the deterministic integral here and the stochastic integral there. So together with James Inglis in 2015, we did the obvious thing. We looked at the sufficient conditions on the connectivity functions for the existence and the regularity of the solution. So we proved this theorem of existence and uniqueness if for all x in Rn, the function y goes to j of x and y is in L1, sorry, and the sup with respect to x of its L1 norm is bounded, then you have existence and uniqueness. I won't go into the details of stated in the theorem, and it's a fairly straightforward application of basic principles in stochastic calculus. Same with the spatial-temporal regularity of the solution. If you add this C3 condition on J, if you take the L1 norm of the difference for 2 points x1 and x2, so the difference j of x1 dot minus j of x2 dot, and if it's upper bounded by some the norm raised to the power alpha of x1 minus x2, where alpha belongs to 0, 1 semi-open, then you have regularity. You have eta 1, eta 2 Holder continuous in time and space. So we're happy we have something we can work with. The final remarks, I'm catching up on time. The final remarks are that probability theory, stochastic calculus, and large deviations, I don't have time to talk about, but Eva mentioned it, are very important tools in mathematical neuroscience and should be put to good use. Consciousness, last part. Consciousness, in the late 80s, consciousness was a tolerated hobby for the aging scientist. I guess that's the class of age I belong to. One of the problems is that consciousness, the word consciousness, is loaded with very fuzzy meanings. You can be conscious of something, you can be conscious. So you have to distinguish between at least 3 meanings: wakefulness, vigilance; attention, focusing your mental resources; and the fact of being able to report what you see, what you hear, what you think. So that's called conscious access. And recently, and most obviously in the work of Stanislas Dehaene at the Collège de France, there are 3 major ingredients that have been made available to researchers to make consciousness amenable to scientific inquiry. So first ingredient is the focus on conscious access, being able to report. The second ingredient is manipulating conscious perception in the lab. And the third ingredient is to carefully record introspection. And of course, thanks to the new quote and unquote brain imaging modalities like functional MRI, EEG, electroencephalography, MEG, magnetoencephalography, or optical imaging, not for humans. So an example of manipulating conscious perception. Here you have a sequence of images. You show either random shapes, which are called masks, or a 33 milliseconds hidden word. Here the word radio, and post-mask, again you mask. And for 33 milliseconds, you don't see consciously the word radio, okay? So if you increase 33 milliseconds to 100 milliseconds, say, then radio will become reportable. You've seen it, okay? But your unconscious has seen it. That's important. And then there's a second example below. Now, what is the unconscious and what is the conscious? I will not answer these very deep questions, but I can follow the end in saying that virtually all the brain's regions can participate in both conscious and unconscious thought. For example, I won't go through all examples, but the unconscious meaning, you can understand meaning of a word without being conscious of seeing the word. So for example, if I show you the word red for a very short amount of time, like in the example before, then this word will prime the corresponding color. By this, I mean that if I next show you a number of colors and ask you to choose one of them, then you will choose with a high probability the red one. So the word red has been understood by your unconscious processing system, and it can prime your action. Okay, so What is the unconscious? Some of you may have read the beautiful book by Henri Poincaré, a French mathematician, published in 1902, and he says that the subliminal self, that is the unconscious, is in no way inferior to the conscious self. It is not purely automatic. It is capable of discernment. It has tact, delicacy, and it goes on and on and on. So the unconscious is indeed very, very, very powerful. Then what is consciousness good for? Why do we need consciousness? Well, at least for these 4 reasons here, it seems that the unconscious is actually doing statistics. So it's keeping, if you have a word like bank, which have 2 different meanings, the bank money or the bank of the river, the unconscious keeps both meanings ready for use. And the conscious, it's only the conscious part of our mind that does sampling. The brain performs some sort of Bayesian inference. It's a very important item. Then the lasting thoughts. Once a piece of information is conscious, it stays fresh in your mind for as long as you care to attend to it and remember it. So that's important when you want to make decisions, for example. The third item is it's only consciously that you can do you can become a human Turing machine. You can do arithmetic. The tape of the Turing machine is just a piece of paper. So the consciousness gives you the power, us the power of a sophisticated serial computer. And it's also, of course, a social sharing device, and there are other examples. Now, Dehaene and his co-workers have identified 4 signatures of consciousness. The first signature is a sudden ignition of parietal and prefrontal circuits. So if I show you words which are masked as before, so they can be visible or invisible, when they become visible, when they're conscious, you have a huge surge of activity as shown as this by this curve in the parietal and prefrontal circuits. So this is the invisible words, not much activity. And this is the visible conscious word. You can report it, and there's a huge surge of activity. Signature number 1. Signature number 2, you have a late slow wave, the P3 wave, which can be detected using EEG. So it uses the same kind of experimental apparatus, and it really corresponds to a huge surge of activity as shown here in the prefrontal cortex. And that resembles very much, a good metaphor for this is a bifurcation in the dynamics of the neural networks underlying consciousness. Third signature, so that you can measure using deep electrodes, like in epilepsy patients, they are implanted, they have deep electrodes implanted, and if they agree, you can use these electrodes to perform some experiments. And again, if you show a masked image and if you increase the delay between the mask and the target image, then it becomes visible to your conscious mind. That implies a sudden and lasting burst of high frequency in the gamma band that is larger than 30 hertz, a huge and lasting burst of high frequency activity. And the fourth signature is correlation or synchronization. Synchronization of information exchanges across distant brain regions. This example here shows, okay, this is a head of a patient with electrodes, EEG. This face is not recognizable, so there is very little correlation or synchronization between the electrodes, whereas when the face is visible and recognizable, then you have a huge amount of synchronization between a large number of electrodes. So these are 4 signatures which have been, and they're very robust, and they have been proposed by a number of researchers, including Stanislas Dehaene and his co-workers. So how do we theorize consciousness? Well, the awareness of a piece of information, by definition, means that it has entered a specific storage area, which makes it available to the rest of the brain. We don't know which specific storage area, but it's there. And it's an old idea. Hippolyte Taines, the French philosopher, talks about the theater of consciousness in the 1870s. He says that he wrote that the conscious mind is like a narrow stage, theater stage, that lets us hear only a single actor, but there are lots of invisible actors in the background which are active. It's an idea also that was widely used in AI in the 1973, so a century later, by Raj Reddy and his colleagues in the speech recognition system hearsay. So processors were using a blackboard, the theater of consciousness, to exchange information. And then, and then the psychologist Bernard Bars, this is the same global workspace, which is indicated here. This is the conscious global space, and you have receiving unconscious processors. And this is also similar in spirit to Jean-Pierre Changeux and Stanislas Dehaene's global neuronal space that they proposed in 1978, which is here, and which receives and outputs information about the past, the present, the future, the value. You can evaluate events, and you can also produce actions. Okay. Now, mathematics and consciousness. This is very, very, very weak, but it's an attempt. It's not original. You can think of a graph G with vertices and edges. Each vertex would represent some brain areas, visual, auditory, somatosensory, and so on and so forth. The mathematical description of this vertex is a set of possibly stochastic neural field equations. Each edge E represents the connections between the various brain areas. The connections can be anatomical through the so-called connectome, collected using diffusion MR, or functional. And this connectivity, these edges may introduce delays. So you have to work about delayed stochastic neural field equations. I didn't have time, but it's a very active research area, delayed neural field equations. And the mathematical changes for you folks are to provide mathematical models of the four signatures of consciousness I just described, such as bifurcations, metastable states, like Effa was talking about yesterday, and so on and so forth. Mathematical challenge is to study rigorously how and when these can appear in the solutions of the neural field equations, and provide mathematical models of the global neuronal workspace. This is an agenda that is going to last for at least a century. Other challenges, computational, already going on, Human Brain Project, The Virtual Brain from Jerza et al., or clinical to study consciousness in people in coma, David Nakash and Stanislas Dehaene. Good conclusion. Very short conclusion. You've all heard about the unreasonable effectiveness of mathematics in physics, put forward by Eugene Wigner in 1960. And to me, you may disagree, that this effectiveness is not yet, hopefully, verified in neuroscience or in biology in general. And one reason for this may be that there are few of the mathematical problems, and some of them are very difficult, raised by these disciplines, neuroscience and biology, that are studied by professional mathematicians. And one of the goals of our community is to bring professional mathematicians to work on, talk them into working on neuroscience problems. This is the end of my presentation. And as I told Eva and the members of the committee, I would like to announce a new journal, MNA, for Mathematical Neuroscience and Applications, which is a bifurcation from the Journal of Mathematical Neuroscience published by Springer, which ends publication this year. Okay, it focuses on mathematics for neuroscience. You can read this here. The most important part, hopefully, is this one: is that it follows the Diamond open access model, which means it's open access, but it's free to the readers and free to the authors. It's free to everyone. This is science for everyone, and this is thanks to the support of episciences.org, which is a non-profit organization. And this is a screenshot of the web page that you can go to mna.apsciences.org, and we are looking forward to receiving your submissions. You can also look at the set of members of the editorial boards. Many of them are on this committee for ICMNS. I thank you very much for your attention. I may answer some questions. I try to stop my clock here, which I followed carefully.
Thank you very much, Olivier, for this beautiful talk. So I forgot actually to announce to everybody that questions, of course, you can ask them like yesterday by just chatting to us, or you can also raise your hand. So downstairs on the Zoom block, there's the possibility to raise your hand if you have any question, and I can unmute you such that you can address directly to Olivier if you wish to do so. So let me just quickly check if there what I should say. So for the moment, I don't see. So maybe I start with the question. So you have been speaking a lot about neural field models all over your talk, also about stochastic ones. Did you consider particular models for stochastic models for these connectivity matrices? And if yes, what would be relevant ones according to you?
In the analysis, I presented the analysis that James Inglis and I did. The function J, the connectivity function, is non-random, is deterministic.
Yeah.
It would be a nice follow-up to our work to suppose that it's stochastic. And I think it would be a good idea. It's more difficult, but it's probably feasible to make some assumptions on J of X, Y, and T as a random process. But we haven't done so. But it's certainly an interesting, valuable research area. Thank you for the question.
So I see that.
Well, the talk was difficult.
Huh?
The talk was probably a bit difficult and fast.
It was fast, of course, because you gave an overview of 20 years of research, or maybe even 30, I don't know. Yeah. So you were also speaking about these, so somehow these long-range interactions in part 2. So it's like a phase transition. So if you add a little bit of them, it completely changes the picture. So
yes.
And if even more increase this small parameter epsilon, is something else happening?
This small parameter epsilon is something which is biologically verified. From what I've read from the literature, it is known that the long-range connections are weaker than the short-range connections, which allowed us to consider the solutions as perturbed solutions to the local homogeneous solutions with the D4 symmetry group. What's important in this part is the role played by group theory. Of course, I didn't have time to go into the very interesting details which can be found in our JMN paper, but I really think that for many sensitivity procedures of the human mind, this group theory approach is important, but also for other things. As I stated at the end, I think group theory is underused in mathematical neuroscience, and I would like to push young people into using group theory in the mathematical modeling of the human brain. It would be also very exciting to combine this with the stochastic aspects, but it's even harder. So
yes. There is a question by Abed Make, but I'm not sure that I understand it. So here's the question. Can you give us your opinion on using information theory to try understand the fingerprints of what? Of connectivity?
consciousness.
Consciousness, yeah, I just didn't have the time to.
Sorry, so information theory to understand consciousness?
Yeah, basically there's this attempt in the field to use information theory to pursue consciousness. Do you think that what you have presented so far, these 4 fingerprints, can we also try to approach, like try to understand them or approach them from the direction of information theory?
Yes, I think so. I haven't talked about information theory in my talk. I couldn't talk about everything, but it is certainly something very worthwhile looking at. Of course, this is connected to notions such as entropy, mutual information, and so on and so forth, which are really natural to use in the area of stochastic neural fluid equations. So there's and also PDEs, of course. But this has not been done. This is certainly an interesting approach, and I would encourage you. All approaches. This is such a difficult and outstanding problem that all mathematical approaches that seem to be relevant should be tried. And probably the answer, if an answer ever comes out, the answer will require a large combination of different mathematical approaches. What's very nice, sorry if what's very nice is that Dehaene, in particular, Dehaene and his group of people have put this, have made a part of consciousness, this ability to report what you feel, what you see, amenable to experiments, well conducted experiments, well understood experiments, and therefore to modeling, because we can now confront our models to experimental results.
Thank you, Olivier. Maybe we have time for one more question. There's one by Romain. So I will read it if Romain doesn't want to ask himself. So brain is characterized by its learning capacities. How would you see synaptic plasticity fitting in the research axis that you described?
Oh, very simply, very simply. The most obvious one is to put some dynamics on the connectivity function J. So that would allow you to incorporate some aspects of learning at this resolution level. So add one more equation to the neural field equations, namely one equation or maybe several equations on the J function, and you can probably account for some plasticity phenomena at this very coarse level. Remember, neural mass is I haven't said it very clearly, but a neural mass is an average of activities over a small volume in the brain. So it's a sort of a mean field approach. So you're already neglecting the activity. You're neglecting the activity of individual neurons. But that may be okay for applications such as consciousness or learning.
Mm-hmm. Thank you very much, Olivier. I think we have to stop here to start your mini symposium in 4 minutes. Thank you again very much.
Thank you. Can you thank you, Eva. Can you tell me what I'm supposed to do? Just stay here?
So, Olivier, if you just stay there, now we are going to invite your first speaker.
Okay.
And from then on, you can share the session. Just you can just call your speakers as you would normally in a room.
Kristin Greenberger, yeah.
Yes, in fact, I was in chat. I was already in touch.
Okay, and all the other people. Okay, perfect.
Should I just starting my screen or?
Yes, you can, I think. And I would just ask Olivier to start sharing the session at 4 sharp so that we are in sync with the...
Even not a cup of coffee.
No cup of coffee?
No.
Okay.
Can you see my slides?
Yes.
Yes. Yes, you can see. We can see it.
Thank you. Great. And I'm also here. You can hear me okay?
Yeah. I'm just trying to write. So the talks are 25 minutes, 30 minutes.
Yes, it's overall 30 minutes.
30 minutes,
including questions.
Okay, including questions.
Yeah. Yes, and if you can read, so I don't know if you can read the chat, Olivier, but if there are questions, I will report them there in the chat.
Yes, I can read.
Yeah, that's good.
So I'm also okay if there are questions that are pressing. You can just interrupt me to answer.
So you don't care about being interrupted. That's okay with you?
No, I don't. I would rather prefer, if there are problems to address them right away. I think that's easier than sort of accumulate them at the end. If that's okay for you.
No, that's fine.
I'm totally okay with it. So
okay.
Last thing I should say, and then I let you get on, is that for those who are watching from YouTube, you can also post your question directly in the YouTube chat, and we will copy paste it here for Olivier to pass it to the speakers.
Wow, that's quite intense. Okay, so we're ready. Welcome to this session, and I will start immediately with the first speaker, Christine Greenberger from Magilab Baylor College of Medicine, and who's going to Brandeis University, and she'll be talking about mechanisms of experience-dependent neuronal computations. Christine, you're on.
Thanks everybody. Let me start by thanking Olivier and Romain for inviting me to talk to you about my work. It's really a pleasure. So just as a heads up, so I'm an experimentalist. I do only very few and very simple simulations myself. But if any of you in the audience, like the work that I'm going to talk to you about today and is interested in a collaboration, just get in touch. My email is on the slide. Okay, so I'm fundamentally interested in understanding the mechanisms that allow brains to produce learned and adaptive behaviors. Now we know that neuronal representations of the external world are formed in the brain by the activity of many thousands of neurons, each responding to diverse environmental features. Now, what determines this feature selectivity is the ability of these neurons to respond to a specific pattern of synaptic input by firing coherent patterns of action potentials. And the work that I'm going to talk to you about today has been testing the idea that experiences that we make during learning change via mechanisms of synaptic plasticity, this input-output transformation in hippocampal neurons, which then enables the formation of new representations that the brain then uses to drive learning. Okay, so to investigate how experiences can change brain activity, I've been studying hippocampal area CA1. And this area is particularly well suited for this line of inquiry because neurons in CA1 form as place cells, spatial representations of the animal's environment, which are both experimentally tractable and known to be involved in tasks that require spatial learning. For example, the very famous Morris-Water maze task. So place cells were, of course, discovered by John O'Keefe and colleagues about 50 years ago. And these are neurons that fire action potential specifically when the animal is in a particular location of its environment. So in this case, I should say that here each red dot corresponds to an action potential and the white line corresponds to the trajectory of the animal in an open field box. So you see that this neuron fired action potential specifically when the animal was in the right upper corner of the environment. Now, in any given environment, about 25 to 50% of CA1 cells will be place cells. Their firing fields, or place fields as they are called, are distributed throughout the entire environment, suggesting that as a population, these cells form a spatial map and inform spatial navigation of the animal. Interestingly, though, when one looks at the activity of a population of CA1 cells, CA1 place cells, it becomes evident that there's more to the story. So in this plot here, this is a population of about 1,100 place cells from my own experiments. Each row corresponds to the mean activity of a single place cell. The x-axis corresponds to the position of an animal on a linear track, and the black color essentially marks for each of these place cells its firing field peak. And you see that, yes, these place cells tile the entire environment, but there's this location here at 90 centimeters with an abnormally increased number of place fields. And so this is, of course, the location where the animals that are thirsty, because they are water restricted, receive a water reward. So what the presence of this reward over representation shows is that CA1 population activity reflects, in fact, the experience of the animal. It reflects what's significant to the animal. And so I'm interested in understanding the mechanisms that allow for this to happen. Okay, so just to orient you briefly about the anatomy. So the CA1 is part of the mouse hippocampus, which is a 3-layered structure located in mice between cortex up here and thalamus down here. It has 3 major subregions, the dentate gyrus, the CA3 area, and the CA1 area. But of course, as I mentioned, we're going to focus for today's talk on the CA1 area up here. From an excitatory input perspective, each CA1 pyramidal cell has 2 major dendritic input compartments: the perisomatic compartment, which consists of the apical oblique dendrites and the basal dendrites, and which receives mostly intrahippocampal input coming from the CA3 area. The other compartment is the dendritic tuft up here, which receives long-range excitatory synaptic input coming mostly from layer 3 of the entorhinal cortex. So in addition to the excitation, there is, of course, inhibition. Inhibition is very complicated in CA1, but I think it's okay to say, broadly speaking, C1 interneurons fall into 4 broad categories. Interneurons that target the axon initial segment, interneurons that target the soma, that target the dendrites, and finally, interneurons that target other interneurons, and therefore are thought to net disinhibit the activity of the principal pyramidal cells. So we know from previous work that inputs coming from CA3 are themselves spatially tuned. So cells in CA3 are also place cells, while it is less clear what the layer 3 entorhinal cortex inputs do. About a fraction of them, about 10 to 20%, have some kind of spatial tuning too, but they are usually described as kind of sloppily spatially tuned. And so now, combining all these inputs that I've told you now in light of these diverse synaptic inputs, the question that I'm going to address in my talk today is really, so what are the circuit mechanisms underlying CA1 place cell activity? Or in other words, what is the relationship between these synaptic inputs and the place cell output of CA1 pyramidal cells? So to address this question in awake behaving mice, I'm training mice to run on a linear treadmill such as this one. You see here the mouse is head fixed. It runs on a track which has a length of 180 centimeters. It's divided up into 3 individual sectors. Each of the sectors is enriched with a different set of somatosensory and visual cues, and you can kind of see it in the video as they pass by. And so this gives the animal the impression to be in a kind of environment while being still in a very controlled environment. So we can easily change features and so on. And so in this case then, as I mentioned, the animals are water restricted. So in this case, I'm giving a reward once per lap, always at the same location. And what I refer to as lap is a complete revolution of the belt or a running distance of 180 centimeters. And you can kind of tell that the animal has learned the task well because of 2 reasons. One, the licking is really only restricted to the location where the reward is given. And the other one is that the animal slows down as it approaches the location where the reward is given, and then accelerates again, and then slows down again as it approaches the reward for the next time. So these are sort of the behavioral parameters I will talk about throughout the rest of the talk: licking and running speed. Okay, so one of the recording techniques that I use are somatic whole cell recordings. So these are I choose to do whole cell recordings because it allows me to study the action potential output of the neuron, of the place cell, but it also gives me access to the subthreshold membrane potential changes, which is of course driven by synaptic activity, which is a reflection of the input side of the neuron. So whole cell recordings are really currently the gold standard for studying input-output transformations in individual neurons. And so to give you an impression what I mean by subthreshold activity, so this is one lab from one of my whole cell place cell recordings. You see the action potentials here in the middle. Here, this is the place field. And so from such a raw membrane potential recording, we can then extract several subthreshold features. But for the purpose of today's talk, I'm only going to talk to you about one, which is this so-called slow membrane potential depolarization in red, or also called ramp. And the ramp essentially describes the fact that as a mouse enters the place field, the membrane potential will slowly depolarize, bring the membrane potential to threshold so that action potentials can be fired. And then as the mouse exits the place field again, the membrane potential hyperpolarizes again, brings it back to baseline. Okay, and so using this technique then, we found a few years back that a certain type of burst firing is really efficient, is really powerful at driving de novo place field formation. So you can see this in this example cell here. Initially in lab 5 or 6, it only fired a few spurious action potentials or was completely silent. But then after firing such a plateau, and I call it a little bit lab jargony plateau, because this is the somatic signature of a large dendritic spike, a calcium spike, originated out in the dendrites of these neurons. So after firing such a plateau, you see immediately after a place field appeared and was there for the remainder of the recording. Now, because we are in whole cell recording, in whole cell configuration, we can then play tricks. For example, we can do current injections to quote unquote, artificially evoke plateaus. This is done here. And you see that also in this case. So this is now not the cell doing it. This is me doing a current injection. And you see that also in this case, a place field appeared and was there for the remainder of the recording. Now, we did current injections everywhere along the track in all different locations. And we found first that it was equally possible to induce place fields anywhere on the track. And second, there was no observable difference in the place field strength, measured by the amplitude of this ramp depolarization that I showed you before. Also, I should say that evoking action potentials instead of these large plateaus was not sufficient to drive place field formation. So it's really a plateau-specific effect. Plateaus are both necessary and sufficient to drive place field formation. And I can also tell you that based on analyses and additional experiments that I don't have time to show you, we concluded from analysis and experiments that I don't have time to show you, that this place field formation was associated with an increase in the synaptic weight and not in the synaptic input frequency. And so what we concluded then from this work is the following: that each C1 pyramidal cell receives a heterogeneous barrage of synaptic input coming from CA3. You see this here, so each Gaussian here represents a single synaptic input. You see they are spatially tuned, and taken together, they cover the entire track. Then after the neuron fires a plateau, there's a subset of excitatory synaptic input that have become potentiated. The result of this potentiation is this ramp depolarization, and as I explained before, then the place field firing. So we also looked at the inhibitory inputs. Unfortunately, I don't have much more time to talk about it, but just to cut a long story short, we found the inhibition to be spatially untuned. So it was very broad, essentially untuned. And so the function of the inhibition in this framework is then to sort of essentially balance out unpotentiated inputs in silent cells, or balance out unpotentiated out-of-field inputs in a place cell to prevent those from firing action potentials. The function of the plateau, of course, is here to essentially select the synaptic inputs that are to be potentiated. And so it essentially determines the feature selectivity. And so the next question that I'm going to discuss with you then are our efforts to understand the mechanism underlying this plateau-driven potentiation. Are there any questions so far, or has it been clear? Okay, then I move on. So to start thinking about this question, what is the mechanism underlying this plateau-driven potentiation? I want to go back to the data. So this is one lab here, down here, where a plateau happened. And then in the next lab, a place field would appear. And so this yellow tick mark up here corresponds to this yellow tick mark down here in position, and essentially marks the beginning of the place field, or you could say the beginning of, or the location where synaptic inputs start to get potentiated. And so I want to point out three features of this process. So once it's very fast, right? Only one trial is enough to drive this potentiation. Second, there's potentiation of inputs that are active seconds before the plasticity driving event, in this case, the plateau. And down here, I marked for you the times in seconds. So you see these inputs here were active 2.5 seconds before the plateau. And the third feature is that there is really potentiation of inputs that were active so far ahead of the plateau that they had nothing to do with driving the plateau itself. And so I'm telling you these features because this contrasts, of course, very much with a established, with sort of the standard model of how we think plasticity works in the brain, which is spike timing dependent plasticity, which in the experiments requires often hundreds of trials, hundreds of pairings. It works only on a timescale of tens of milliseconds. So you need a tight temporal relationship between the pre and the post-synaptic activity. And third, there's a directionality to the input. So only those inputs that were active before the postsynaptic cell was active are to be potentiated. And so we were very excited about this finding. And so we wanted to explore this further, especially because it was so different from the standard model, how plasticity works in the brain. So the first question was, okay, it seemed to have a long time course, but what is the exact time course of this plasticity? And so to address this question, Aaron Milstein, a former colleague in the lab, who's now a professor at Rutgers, and Sandro Romani, who's a computational neuroscientist and group leader at Janelia Research Campus, took our data about the extent and the amplitude of the ramp depolarization, and they made a model. And so the plasticity kernel then that best fitted our in vivo data is here shown in white. And so this is contrasted here with the plasticity kernel for the standard spike timing dependent plasticity rule in red, which has a time course of about 50 milliseconds, width of about 50 milliseconds. And so you see that our plasticity kernel is about 2 orders of magnitudes longer. It extends all the way from 3 seconds ahead of the plateau up to 1 second after the plateau. So to summarize here this slide, the plateau-driven plasticity seems to follow an asymmetric seconds-long rule. And so based on this finding, then Sandro came up with a prediction, namely that if this is true, if really place fields are formed according to this new plateau-driven plasticity rule, then there should be a linear relationship between the width of the place field and the running speed of the animal in the induction lab. And to illustrate what we mean by that is, let me show you 2 extreme conditions. So first, if the animal is running very slowly when the plateau happens, then only a few synaptic inputs are active within that 4-second plasticity time window that I mentioned before. So the consequence is only a few inputs will be potentiated and the ramp will be very narrow. In contrast, when the animal is running very fast, when the plateau happens, then a much larger amount of synaptic inputs fitted to that same 4-second plasticity window. More inputs will be potentiated and the ramp will be wider. Okay, so we looked at our data. So I'm showing you here on the x-axis, on the y-axis, the width of the ramp, place field width essentially. And on the y-axis is the running speed during the lab when the plateau happened, during the induction lab. And you see that indeed, there is this nice linear relationship with a plot with a slope of about 2.5 seconds, very much consistent with our 4-second plasticity rule. And the red dotted line shows you the slope that we would expect if SCDP would apply, because essentially there would be no slope there, right? So in a 50 millisecond window, there are very few inputs are potentiated. So there's really none of these relationships. So our experimental data really supports this idea that place fields are formed according to that plateau-driven plasticity rule. And just to summarize, then, so we think this new plateau-driven plasticity rule is really a new form of synaptic plasticity that we discovered. We call it behavioral timescale synaptic plasticity, or BTSP, and it has the following features. So it is driven exclusively by these plateaus that I mentioned, are actually the somatic signature of dendritic spikes, calcium spikes in the dendrites. It's a true one-trial learning rule, so a single plateau is enough. I didn't have time to show you the data, but we have evidence, and I'm happy to talk about it if there are questions about it. But we have data that it affects the proximal synapses, the CA3 to CA1 synapses, and it follows this asymmetric seconds-long time course.
Christine?
Yeah.
Christine, there is a question on the chat. Would you like to hear it? It's a question by William Brown, who's asking if is the ramp a filtered membrane voltage, or are there no spikes at all? Question mark.
Sorry. I should have mentioned this. Yes, so the ramp is essentially taking the raw membrane potential recording, cutting off the spikes, and then low pass filtering it below 1 Hertz. Yes. Okay. Sorry, I should have been more clear about that. No, it's essentially every time you have a place field firing, you find this ramp that underlies it. So this is, Thanks for this question.
Fine, thank you.
Okay, so you might have noticed, and it's good that we talk now about the ramp, because you might have noticed that that in the example I showed you, the ramp is kind of asymmetric actually. And this is of course due to the asymmetry in the plasticity time course. And this has in turn an interesting functional consequence for the CA1 output, namely that place field firing that comes out of the CA1 area always precedes the plateau that generated it. So in this case here, you see here's the place field firing. Here, this is the plateau. And you see that, in this case, about an average, this is about 10 centimeters. The peak firing precedes the plateau. So we conclude from this that BTSP, in fact, produces place fields that are predictive of the event that generated it. And I find this very interesting to think about what could be the behavioral consequence of this. Okay, so I just added this now to the list of features. And so in the final part, I want to talk briefly about some unpublished work. So based on these features, then BTSP is really a mechanism that could allow neurons to store an association between an event and a delayed outcome, let's say a reward, right? And that outcome can be delayed by up to a few seconds. And so because of this unique ability, then my idea was, okay, could be actually BTSP then the mechanism that allows for this reward over representation to form, that allow for this experience-dependent shaping of the CA1 population activity to form. And so then the last question that I'm going to talk to you about today is then, is BTSP actually the mechanism that allow experiences to shape CA1 representations? And the plateau could be kind of seen as a kind of teaching signal, saying, okay, something significant has happened, now form a place field here. And so to answer that question, I turned to a different technique, which is 2-photon calcium imaging of CA1 representations. This is the timeline of my experiments. So the idea is that after a surgery that allows us access to the hippocampus, a short training period, then I image the same population of neurons for about a month. But for the purpose of today's talk, I'm only going to talk to you about the first 2 days, which I call day 0 and day 1. And so what's the difference there? So day 0 was done in contrast to what I showed you previously on a belt that is devoid of any cues, and you have variable reward location. So this is essentially only an adaptation phase for the animal. And then day 1 is on a cue and a belt that is enriched with cues with 1 fixed reward location. It's very much like I showed you before. And you see that this is now the licking behavior. So each of these ticks marks a lick. And you see that here on day 0 with the variable reward locations, there is no really pattern. There's no pattern. The animal essentially licks everywhere. But here then, after initially licking everywhere, the animal realizes, okay, I have to restrict my licking more and more to this specific reward location. And we can quantify this it essentially happens in all the animals. So this suggests to us that, yes, we can essentially make a connection between our behavior and learning. And now we, of course, want to look at the activity of the neurons during this learning behavior. So this, just very briefly, so this is how an experiment looks like. You see here a typical field of view with CA1 pyramidal cells. On the right side, you have a activity. So this is using a calcium indicator called GCAM6F. And you see here on the right, 9 place cells, the activity. On the bottom, you have the position trace. So as the animal runs, the position can be read out. And so based on these movies, then we can extract the activity for each of these place cells. Mm-hmm. So this is then the color plots that I generate based on that data. You see for this I've shown you essentially before. This is now just data from a single animal. You see this accumulation of place fields as 90 centimeters, which is the reward location. And in contrast, on day 0, there are much fewer cells and their place fields are much sloppier. I can quantify that. So this is data from this one single animal, and this is data across all the animals I did this experiment in. And you see that already on day 1, you have this reward over representation. So within an hour of experience, within an hour of doing this task, you have this reward over representation forming. And because all of this seemed to happening on day 1, I can then ask a question about the time course. So these are 2 example cells. So you see that essentially cells are initially silent, and then just sort of out of the blue, you have these place cells popping up, very much reminiscent of what I showed you before with the whole cell recordings. And so this is the time course of the appearance of these cells. You see that there is an initial phase, maybe 30% of the cells appear really early on, and then the majority of the cells sort of appears gradually over the session. But of course, the question is Btsp involved? And so I have different lines of evidence that say, yes, it is. I'm trying to show you only once, which is I can pharmacologically block plasticity, and I can pharmacologically block dendritic calcium spikes. And in both cases, essentially this reward over representation does not form. Okay, so this is shown here in the red traces. So to summarize then, what I've told you so far, so CA1 has 2 major inputs. Potentiation of the CA3 to CA1 inputs via Btsp, counteracted by spatially uniform inhibition, leads to predictive CA1 output and an output that is experience dependent. Now, very last point. So of course, the plateaus have this really important role. And the question is now, what drives these plateaus in vivo? And so we have previous experimental evidence that plateaus are actually driven by entorhinal cortex activity. And so I wanted to essentially ask the question, can we look at entorhinal cortex activity? We can image those axons that come into CA1 and see whether they could be responsible for driving these plateaus. And long story short, this is essentially what we found. So this is the activity of about 150 axons that I imaged with using a calcium indicator. And you see there is this peak here around the reward side. This is the average activity. Okay, so then to really summarize now. So this is the picture that emerges for us. So we have a neuron here that produces an experience-dependent and predictive output. We have 2 pathways. We call the EC3 input a pathway, the instructive pathway input, because this is the pathway that drives the plateaus. And then you have the CA3 input, which is essentially the learning pathway, because that's where Btsp works on. And so this is the instructive signal then. And so I didn't have time to talk about this part at all, but we think that essentially what determines which synapses undergo plasticity is determined by a so-called eligibility trace, which was mentioned yesterday in a few talks, which is a filtered signal of the synaptic input. And so the combination of this instructive signal, which is the plateau, and this eligibility trace in certain synapses then lead to these weight changes. And this is just to remind me to mention that there's other work from other labs showing this inhibition has a permissive role as well as neuromodulation. So it's really a sort of all the circuit elements form to work together to kind of drive Btsp. These are the people that contributed to the work. I want to thank especially my postdoc advisor, Jeff McGee, who's been just fantastic. As I mentioned, as Olivier mentioned, I'm going to open my lab essentially in 2 days. So if you are interested in joining or in collaborating, come visit us at beautiful Brandeis campus near Boston. So this is Boston downtown. You have the Charles River coming by. It's really worth a visit. And get in touch via my email, or this is the webpage. Thank you. And sorry for going over time a little bit.
Are you over time? I don't think so. No, you have 2 more minutes. Let's see. You have one more question from, several questions.
Right.
All right. So, question by William Brown again. So, about the first part of the talk, are the plateaus always caused by dendritic calcium spikes from the apical tuft, or can they also be caused by sodium spikes that might be caused by CA3 input? Question mark.
Yeah, so let me just, so plateaus are always caused by these calcium spikes. So there is slice work from Jeff's lab and other labs. And so essentially what marks this calcium spike is this large depolarization that underlies the small sodium spike. So this is really the signature of those. So they are always associated with calcium spikes.
Okay, a question by Romain Veltz. Do you think this plasticity rule is implemented as regular quote end of quote LTP, AMPA insertion, or uses another mechanism?
Yeah, so this is totally speculation now. We are working on that, but yeah, that would be the idea that there is AMPA, there is rapid AMPA insertion going on within a few seconds after the plateau. So there might be some pool of receptors, right, that are readily inserted.
Okay, in a few seconds. More. Okay, I'll have to skip. Do you think that this type of plasticity generalizes to other types of tasks by Einberg?
Yeah, very good question. I think, so, I mean, it's not only to tasks, but to other areas. So I think it would be useful for many different tasks in many areas to have this sort of long timescale plasticity rule. But we don't know yet, right? So we welcome everyone to, try these protocols and see whether in their favorite task or in their favorite brain area of interest, you see a similar task. I should say that these plateaus are not specific to CA1. Any pyramidal-based microcircuit has these dendritic calcium spikes. So in principle, also layer 5, layer 2, 3. So all these circuits have plateaus. Whether they cause plasticity, is another question.
Thank you very much, Christine. We are over time. There are also more questions. You can look at them and answer to them.
Yeah, absolutely.
In the chat. So we're now moving to our second speaker today, Gaëtan Vignoux. Daniele, can you put up his screen?
Yes, I'm doing it.
Gaëtan is here. You start sharing, maybe.
Yes. Do you see my slide?
Yes, I do. I hope the others can also. So this is the talk by Gaëtan Vignoux, joint work with Philippe Robert, so INRIA and the talk and Collège de France. And the talk is on the spontaneous dynamics of synaptic weights in stochastic models. Gaëtan, the floor is yours.
Okay, thanks. So I'd like to thank the organizer for inviting me to talk in this symposium. And so I will briefly present some work from my PhD, which I'm doing with Philippe Robert on the implementation of spike timing dependent plasticity in stochastic models of neurons. So I'll go quick on the introduction because everyone here, I guess, know about neurons. So in my work, we only consider a simple system where we have a single neuronal cell, which is connected to a second one, so the green one, through a single synapse. So it's a really minimal system to study the influence of plasticity on this synapse. So I will just remind here that we consider that the information is going unidirectionally. So it starts from the presynaptic neurons, goes through the synapse, and then go to the postsynaptic neurons. So I will use the term presynaptic neurons and postsynaptic neurons in the following of the talk. So the information goes through spikes, which are initiated in the first neuron, the presynaptic one. And then at the synapse, it gives place to a rise of the postsynaptic neuron membrane potential, so a short rise, which can afterwards lead to a spike of the postsynaptic neuron that is then propagated to the rest of the system. So we are interested in our work about synaptic plasticity, which is the modulation of the influence of the presynaptic spikes on the membrane potential jumps at the synapse. So for example, here I represented a potentiation, which is an augmentation of the jump size after some activity-dependent process, like the one that has been presented just before by Christine. And so it is quite known that this is the basis of learning and memory in the brain. So our approach is at the junction between three different domains. So firstly, we use experimental data from neurophysiology, so from my lab at Collège de France, about STDP in vitro and in vivo. Moreover, there have been a lot of computational neuroscience study on the role of STDP in numerical networks, so both bifurcation analysis, but also just computer simulation and more complex networks. And finally, we use the fields of probability to study rigorously how does STDP evolves in stochastic neural networks. So up to now, there have been only in the probabilistic field, stochastic neural networks have fixed weight. And so our goal is to implement a stochastic neural network with weights that are modulated by the network's dynamics. So that's what I will present today. I will present how we implement STDP in a stochastic setting, and I will present results on pair-based models in that case. So I'll start by the general framework for STDP that we have defined. So to go back to our simple system, so with just two neurons and one synapse, we represent the presynaptic neuron by an homogeneous Poisson neuron, so whose spikes are represented by the jumps of a Poisson process with a fixed and homogeneous rate, which we call lambda. This leads through an integrate, leaky integrate dynamics to this kind of membrane potential traces, so which is just the convolution of the presynaptic spike train with an exponential kernel. So we don't use any reset or other properties of internet and fire neurons. And then, and so the jump size in the convolution is what we call the synaptic weight. So it will be the value that is modulated by synaptic plasticity. We call it W. And so the postsynaptic spike train is modeled by, again, a Poisson neuron, but this time its rate is proportional to the membrane potential X, which is the yellow trace. So in mathematical term, what we call saying that the presynaptic neuron is an homogeneous Poisson process with rate lambda, it means that if we just take the probability of having a spike in a small time interval delta t, the probability is equal in the limit of delta t going to 0 to lambda times delta t. Then to model the postsynaptic membrane potential, it's just a short noise, an exponential short noise of amplitude lambda. So it's just, it follows the following dynamics. So it's almost the same than in the leaky internet and fire model. And so the postsynaptic spikes, the probability of having a postsynaptic spike in a small time delta t is equal to beta, which is the activation function, usually an increasing function of the membrane potential x at time t. So like this, the higher the membrane potential, the more the more probable it is to have spikes of the postsynaptic neuron. So then how do we model synaptic plasticity? So it's the new the addition we have done. So synaptic plasticity, as was explained also before, there are many models, and one that is really used in computational science is STDP, because it's only based on the presynaptic and postsynaptic spikes, which is really then easy to implement either in mathematical analysis or in computational works. So this is a classical Hebbian curve from B and Poo work. So here you have delta of t, which represents the timing between the postsynaptic spikes and the presynaptic spikes. And here are relative synaptic changes after a protocol of several pairings. And so here you have on the right, so it corresponds to when you have a presynaptic spike that is followed by a postsynaptic spikes, you have potentiation. And here on the left side, which corresponds to a delta of t negative, so it's the anticausal pairing, where the postsynaptic spike is before the presynaptic spikes, you have depression. So starting from these classical experimental curves, we just take an exponential model. So where we model, so as you see, we just fit an exponential curve. So with two parameters, the decay time and the amplitude at 0. And then we want to model, to add this to our model. So here I represented one presynaptic spike and 5 postsynaptic spikes, which is below. And so how do we update our synaptic weight, which is below? We just update at each postsynaptic spike time, so t post, so the green one, the purple one, by the sum over all previous presynaptic spikes. So here there is only one. So we have two terms in the sums. Of the quantity B1 over exponential minus gamma 1 t post minus t pre, which corresponds to the red dots that you have here. So it gives us this sum, which can be modeled through the following short noise processes. So Z1 is the sum of B1 exponential minus t pre. So it's a stochastic process that at each presynaptic spike, so the green one, is raised by the value B1 and then decays experimentally by the value gamma 1. So gamma 1 is has been forgotten here, but it should be here. And then we update W at each postsynaptic spike time by the value Z1 at time t post. So it gives the same equation. It's just a more convenient way to represent it in terms of Markov processes. And so here is the differential, the stochastic differential equations associated. So the first one is just a classical short noise processes, and the other one, it just means that at each spikes of the postsynaptic neuron, we update by the value of Z1 just before the spikes. We can do the exact same analysis for the other side of the curve, so the blue one. So at each presynaptic spike t pre, so the green one, we update by the sum over all previous postsynaptic spikes, so the 3 purple ones that you have there, by the quantity B2, which is the amplitude at 0, exponential minus gamma 2 t pre minus t post. And so again, we have the same representation in terms of stochastic differential equation. And so all in all, this gives us a simple system of 3 stochastic differential equations. So the first one, which represents the activity of the presynaptic neuron. The second one, the activity of the postsynaptic neurons integrated on a time scale. And a final equation where we have the update of the synaptic weight in terms of this value of Z1 and Z2. So here I presented the all-to-all implementation of STDP, which means at each spike, I just took into account all the previous spikes of the other neurons in the sum. There have been a lot of work to compare and to try to find the best way to model STDP. And so other implementations have been developed. One of those being, for example, the nearest neighbor symmetric implementation, where for each spikes, we just take into account the previous one. So as you see, I have lost 2 terms in my sum because these 2 ones are not taken into account anymore. And there is also a more restrictive one, which is the reduced implementation, where we only take into account consecutive pre and post pairing. So which means we don't take into account this one because there is a postsynaptic spikes between the previous presynaptic spike and the current spikes. So we only have two terms in this final implementation. And so we can also, by the same tricks as I explained before, represent this process with more complex system of stochastic differential equation, which I won't present here. Just let's say that they exist and we can show that they're equivalent. So what we have done in our work is to suppose, to make an hypothesis that has been used by most computational neuroscience article on this subject, which is that there are two timescales in STDP. There are fast processes, which are on the timescale of milliseconds, which are mostly the timescale of the membrane potential dynamics and of the correlation in the STDP curves. So that's the one Christine also presented before, so a very restrictive one, around 20 to 50 milliseconds. So it's really, really short. The other processes, which is the updating of the synaptic weight, so the characteristic amplitude of change of the synaptic weight, it takes more times because protocol, for example, they do a protocol on about like 1 minute, and then we have to wait for at least 5 to 10 minutes to see a significant change appears. So the changes in W can be characterized as slow. And so what we do in mathematics when we have this kind of two timescale system, we use the scaling parameters to uncouple these two systems. So that's what we have done. We have developed a formalism and introduced this limit epsilon when epsilon goes to 0. And I will just present the stochastic averaging principle that we have proved, and then I will use it in an example. So what we can imagine is that as the synaptic weight W is evolving really more slowly than the neuronal dynamics, is that we can suppose that the neuronal dynamics happens with the synaptic weight fixed at a fixed value W, and then we can study so this stochastic differential system with a fixed W, and we can study the equilibrium if it exists, and what's the property of this equilibrium distribution. So we can prove that for simple systems like this, which are just like multidimensional short noise processes, the invariant distribution exists, and that the system converges to this distribution really quickly. So I will note by pi of x the invariant distribution of this stochastic system with W fixed. So the indices W here is for the value of W used in the system. And X can be either of the three schemes I presented before to implement STDP. And so the idea is that when we look at the system from the point of view of the slow variable, so W, the system has the time to average over itself, because the neural dynamics are going really more quickly than synaptic weight ones. And so we have the following equations in the limit of epsilon going to 0. So I will present it. So firstly, for mathematical reason to avoid any explosion in our system, our proof is stated with the existence of a finite or infinite horizon S0, which represents the time on which the limit system is defined. So we have proven that there exists an S0, which can be finite or infinite, where the stochastic process W of epsilon is tight when epsilon goes to 0. So which means compact in topological term for the convergence distribution, and that any limiting point small w of t satisfies the following equation. It's kind of what we could expect from intuition. It means that the synaptic weight is equal to the integral over the invariant distribution of the quick system at W fixed of polynomial terms in a way of Z, Z1, Z2, and X. And so what will be interesting for us is that with the simple system I presented you before, we can compute this and have an explicit equation in the limit of epsilon going to 0. So I will now present the result for a really simple case where the activation function is taken as linear. So the form is like nu plus beta times x. So nu will represent external output, and beta times x will represent the spiking process due to the presynaptic input. And so to study how the system evolves in the limit of epsilon going to 0, we will need to study the functional that I presented before, which is the integral over the invariant distribution of this term. So we will be able to compute it for all the model I presented, and then to study the dynamics of the equation. And so I will also present simulation for different types of STDP curves and do a bifurcation analysis. So for the first model, the computation of the function f leads to a really simple equation where we have the derivative of w, which is equal to just an affine function. So where we have like a term multiplied by w plus a constant. So the terms before that depends on the constant of the model. So I just remind you, B1 is a pre-post amplitude, B2 is a post-pre amplitude, gamma 1 and gamma 2 are the time constant of STDP. And so we have just some terms like this. And so from this equation, we have two possible behavior when nu is equal to 0, which is the case where we have no external input. Either this term is positive, is negative, and in that case, we just have the derivative being a negative function, and so the w converges to 0 when the time is going to plus infinity. So that's what we have here. I just plotted some simulation with different epsilon to show you that when we have a curve where the term lambda 1 plus lambda 2 is negative, we have convergence to 0 for asymptotics. Then, if we have a positive terms below, we have the other situation. So w diverges to plus infinity when the time is going to plus infinity. So I just represented here like a summary of this, where we test all the value of B1 and B2, and we show that if we are below the dotted line, we have depression, so the synaptic weight is going to 0. And if we are above, we have potentiation, so the synaptic weight is going to plus infinity. It's kind of a simple behavior, and it's not really interesting because what we want to have are stable fixed point or unstable fixed points to have more complex dynamics. And so to do this, we just have to add an external input. If we add an external input, this term is not 0 anymore. And so depending on the sign of lambda 1, we can have either a stable fixed point like this, so where we have convergence of our synaptic weight to a positive value at high times. Or if we just reverse the sign, we can have the other way around. So we can have an unstable fixed point. So if we start below this fixed point, we go to 0. If we start above, we go to plus infinity. And so here is like the summary map where we can see that we have two small regions. So between the dotted lines and the plane lines, if we are here, so in green, it's stable fixed point. If we are below, in orange, it's an unstable fixed point. And if we are outside of these two, we just go back to the simple behavior as before, which are convergence to 0 in blue, or divergence to plus infinity in red. So now if I go to the second model, so the nearest neighbor symmetric pair-based rules, so it's the one where we only take into account the last spike of each in the sum. In that case, the computation is more complex because it's not as easy to do the computation, and we have a really more complex function, so the dynamics is not as easy to study, but still we are able to prove that we have the following map, where again we have 4 zones, one of LTD, so where we have only convergence to 0, which is again in the same region as before, LTP, and we have a stable fixed point zone, which is way larger than the one I presented before, which was like this, way larger, and the same for the unstable fixed point. So this was done for no external input, and if we add an external input, it's kind of the same. we really don't have much changes. And finally, what's interesting is that if we look at the last model, which is the reduced one, where we only take into account consecutive pairs of spikes, we have a switch of stability, which means that where we had before stable fixed points, we have unstable ones. So it's just like the orange ones. And my figure is not really detailed, but we can prove that we have, if we are close enough to the plane lines, we have the existence of an unstable fixed point. And so symmetrically, in these regions, we have the existence of a stable fixed point. So it's really interesting because in many computational studies, all these pairing schemes are used without any justifications. And here we show that in the limit of low external input, so when we have small external inputs, there is a strong impact of the pairing schemes on the dynamics of a single synaptic weight. It can lead to either stable or unstable fixed points, depending on which scheme we choose. And it's kind of important then when we want to study afterwards dynamics of neural networks, when we do this choice of pairing scheme, to justify it and to have in mind that it can lead to opposite behaviors. What we have also showed is that if we add a significant external input, then all pairing schemes are equivalent in the sense that the stable and unstable fixed points happen in the same regions approximatively. What we have also shown is that to have a stable fixed point, so we need anti-Hebbian STDP, which is not the most well-known one. So where we have depression on the pre-post sides and potentiation on the post-pre sides, whereas Hebbian STDP leads to unstable fixed point. So many studies have been done on STDP in networks, but mostly with Hebbian STDP. And so it will be interesting to study how anti-Hebbian STDP evolves in more complex network. And so to finish, I will just say that we have also done this work for inhibition. So like I presented here with positive synaptic weight W, but we have also done it for negative ones. And so the situation is reversed. And we are planning to extend this model to multidimensional models and to study the plasticity in complex neural networks. And we also plan to do some limits for large network with mean field approximations. So these are the associated papers which are in my PhD. And so I thank you a lot for your attention. And I'm standing by for any questions.
Thank you, Gaëtan, for this very interesting talk. I'm looking at the chat. I don't see any burning questions so far. Could you perhaps tell us a little bit more about the mean field approaches that you're considering, and perhaps the kind of scalings that would be appropriate, you think?
Yes, so it's just like really preliminary works. What we are thinking is that we may not go as many other people have gone, which means by taking the mean field limits on the presynaptic inputs, because usually what people do is that they still keep like a feed-forward architecture with many presynaptic inputs and one postsynaptic output, and they do the mean field limit on the presynaptic inputs. But I think we will more go to the situation where we have many postsynaptic neurons and do the mean field on them. And to do this, we will just scale the interaction between these postsynaptic neurons, because we will have an infinity of neurons and they need to be connected. And so we will scale by 1/n their interaction strengths.
Okay. There's a question on the chat by Sven Gödicke. Very interesting, he says. Can you also say something about the fluctuations of the weights or the weight?
So we haven't done any central limit theorem on our convergence processes yet, because we haven't done it. But I guess we could expect that in some limit it scales back to Gaussian processes. But I'm not sure which limit, and I don't want to say any missed many wrong things, so I won't say much more.
Okay. Okay, thank you. I've lost the chat. There it is. Yes, Abe Make, any intuition on the diffusion between the symmetric and the pair symmetric to see when the switch happened between stable and the unstable regimes?
So we don't have like a continuous parameters that goes from the symmetric model to the reduced one, but I guess we could try to find one and to study more precisely this switch. This is a really good question, and I would be really interested in doing this because I think it's really important to have this in mind. But as I said, we also have a switch when we raise the external input, but this will be more easily studied, and we are going to do it in the publication, I guess.
I mean, is it just because, like in the symmetric one, you include every spike after the green spike? So in the post-synaptic ones, so basically, if you reduce the neighborhood, maybe this is how you can make your diffusion, make it continuous by reducing the nearest neighborhood on the post-synaptic one, probably. Yeah. Anyways, just.
I don't see how I can have a continuous parameters to scale from the symmetric one to the reduced one. But if you have any ideas, I would be happy to talk about it in the chat or even later by email.
Okay, that's a starting collaboration, perhaps, with Abed. If there aren't any more questions, I will thank the speaker, Gaëtan, and thank you very much for this nice talk. And we'll have to go to the next presentation, which is by Pascal Henson.
Exactly.
Pascal, are you here?
Yeah.
He's here. Can you start sharing?
Yeah, I can.
So Pascal just finished completed his PhD under Romain Wels and Etienne Tenré at INRIA. And he's going to be talking about slow, fast, and long-time behavior analysis of a neural network with stochastic STDP.
Exactly. Do you hear me well?
Yes.
Okay, so let's go. So hello, everybody. Thank you. I would like to thank the organizers for enabling me to talk about my joint work with Etienne Tenré and Romain Wels. So on slow, fast, and long-time behavior of a neural network with a stochastic STDP. So it's a bit related with the work of Gaëtan. And I will first give the motivation of of our work, and then give the presentation of our new model, and go on to speak about our mathematical results and illustrate them before giving perspectives. So our work, in our work, we wanted to use mathematical modeling and mathematical analysis in order to get more insight in the interaction between the dynamics of the synapses and the dynamics of neurons. So there exists a few mathematical results, as Gaëtan said, on plastic neural networks. The main reason is that because it's quite difficult, because it involves the coupling between the dynamics of the neurons and the dynamics of the synapses. And one way to simplify such model is to use the timescale difference between the dynamics of the synapse and those of neurons. And it was first done by Kempter and his collaborators and his colleagues. And it follows a lot of numerical analysis. And since a few years, there are more and more mathematical results on the plastic neural networks. And for instance, I just gave 3 of them. So the work of Galvez and his collaborators on short-term plasticity and using mean field methods. Torres and Salor studied neurons that are modeled by time-elapse models, and they study the long-time behavior of this neuron interacting with synaptic weights that are plastic. And finally there is the great work of Robert and Gaëtan that he explained us just before. So in that context, we proposed a new model with a stochastic plasticity and discrete synaptic weights. This model has we proposed it because to have a biologically realistic model, and we wanted it to be mathematically tractable, and also we wanted a model that we can simulate with a large number of neurons. And we then studied it using biological assumptions, mainly this timescale difference, and this enabled us to do slow-fast analysis, and we then obtained a reduced model that we then studied its long-time behavior. So I will first present briefly, because it has been presented before, the STDP, so the spike timing dependent plasticity mechanism. So if we consider two excitatory neurons, we want to know how the weight Wji will evolve depending on the spiking times of the neuron I and J. So what has been observed is that when the presynaptic neuron I spikes will cause repeatedly the spikes of the neuron J, we'll observe an increase, so long-term potentiation of the weight Wji. And in the other way, we'll observe a depression of, so a decrease of this weight. And this has been studied experimentally, and it has been shown that there is an interesting dependence in between the spike timing, so this delta t, and the change of the synaptic weights. Well, another thing which is interesting is that we need a lot of spike pairs before observing a significant synaptic change, so synaptic weight change. So we can model this phenomenon by two ways. So whether we consider infinitesimal increments of synaptic weights each time we have a spike pair, so yeah, it spikes, or we can, so this leads after a large number of spike pairs, this will lead to a macroscopic change. This is done by Kempter and the following studies. And another way is to consider macroscopic change each time we have such a spike pair, but this jump will have a small probability to happen. So this is the way we model the plasticity, and this leads to stochastic plasticity and to discrete synaptic weights. And this seems to be more biologically realistic, because synaptic weights should be mainly discrete because of the number of chemical and molecules that are responsible for this plasticity, so for the synapses. So this has been shown by O'Connor. So now I will present you our new model. So we use the stochastic Wilson-Cowan model for the neurons, in which we added the given STDPs that I will describe. So we consider a network of n interacting neurons that are described by their potentials that are binary and that will be noted by Vt. And these neurons are connected by the synaptic weight matrix that will denote by Wt. So this matrix is it takes integer value, so we will consider only excitatory neurons, but some of our results hold also if we consider inhibitory neurons. I will tell you which results still hold. So I will present now the dynamics of the weights. So our weight will change as soon as one neuron spikes. All its incoming and outgoing weights have the possibility to change. So first, all the incoming weights will have the possibility to increase with the probability P+, which will depend on the last time, the last spike of the neuron. So in that case, the neuron J. So we see that this variable S will correspond to the delta T that we presented in the experiment of B and Poo. And so in the other way, the outgoing weights will have a probability to decrease, which still depend on these times in the last spikes. The function P plus and P minus, P plus and P minus are taken to approximate the results of B and Poo, or you can also take any functions, because we know that there are a lot of different STDP curves. So in this example, I show you with the approximation of the experimental the experiment of B and Poo. So now we have to add this ST in our variable, and also we need to describe the moment that we consider to be a spike, and this moment will be the jump of the potential from 0 to 1. So I show you here in the graph that when the neuron 2 jump from 0 to 1, we will just reset the corresponding S to 0, and etc. So we add in the dynamics that when the neuron I spike, the corresponding S will be reset to 0. So now we have to describe the dynamics of the potential, and these dynamics will depend on so the spiking rate, so the jumping rate from 0 to 1 is a function of the synaptic current entering into the neuron I, and the jumping rate from 1 to 0 is constant, because we want to model the time of a spike, which is quite constant in the biology. And one last thing is which is important, is that we consider that the spiking rate is bounded, and yeah, also the alpha curve is can be considered to be the sigmoid in general. So now I will just give you a last example to be sure that the model is clear. So if we consider a 3-neuron network, we can just now show how it can evolve when the neuron, for instance, the neuron 1 spikes. So we see that just before the spike of the neuron 1, the network was in that state. And when the neuron 1 spikes, so you will have the jump from 0 to 1 to the neuron 1, and the jump of the corresponding S so the reset to 0, and you can have a change of the weight. So the outgoing weight can decrease. So for instance, this one has decreased, this one did not decrease, and the incoming weight can increase. So now we have presented this model. We want to study it mathematically, and so it's we cannot do it with such a model because it's quite complicated with this dynamic on the synapse and this dynamic of the neuron on the neurons. So we will make the assumption that the plasticity is really slow compared to the dynamics of the neurons. So we will introduce a parameter epsilon, a small parameter, that will be used mainly in the definition of the function p plus and p minus. And we will assume that now the probability is epsilon p plus and epsilon p minus, the probability of jump of the weights. So we see that now we will be interested in the dynamics when epsilon tends to 0. So we see that this process, this adding of epsilon will make the weight be fixed in that limit. So the first question we can ask is, does the process Vs has an equilibrium for when the weight matrix is fixed? And then we can study the dynamics of the weights on the fast scale, and then we will obtain a non-trivial dynamics of this of the weight on this time scale. So first we need to spare the state space of our process. So we'll consider that E1 is the state space of Vs and E2 is the state space of the weights. So the first result we have is to show that when W is fixed, so the synaptic weight matrix is fixed, we can show the convergence of the process Vs to a given invariant measure that will depend on W, and this occurs exponentially. This result is shown using the criterion, the doubling criterion. And after that, we then study the process W on the faster time scale T over epsilon. And we show that this process will converge to a process W bar, which is a pure jump process that is defined by its jumping rate between W with that can be jumps that can make the weight jump from W to W plus Eij or minus Eij, where Eij are is the canonical basis of the square matrix, the n square matrices. So what is important is that this rates Rij will depend deeply on the invariant measure Pw. And so now we have these dynamics. We wondered how we could study the long time behavior of this limit process W bar. And we can, to do so, we can use the so-called Foster theorem. And with this theorem, given the Lyapunov function, we found that we can find condition on the jumping rates of the W bar, such that the process W bar will converge to a unique invariant measure. We have the same results for divergence, using the fact that instead of this condition, we will have this left term that will be lower bounded by alpha big M, capital M, n square. Just to come back to the assumption, the first one is just to be sure that our process is irreducible and aperiodic. So this is a nice result, but we still need to compute this Rij and plus and minus to be able to use it. So to do so, we use the Laplace transform of PW that appears naturally in if we write P plus as exponential, if we consider P plus to be exponential. So this is where the Laplace transform appears. And we can also have a similar result. So you use this Laplace transform in more general cases. So if you can write P plus and P minus as a Laplace transform of other functions. So now with this results, we are able to compute the Laplace transform of PW in the case of only 2 neurons, and we can in that case. So I didn't put all the parameters, but I took general parameters, and we can see that we have the convergence that we have proved. So this is for so for epsilon equal to 0.01, and for 10,000 trajectory, we obtain that the distribution at after 3 seconds is quite close between the particle is the initial system and the Laplace is the W bar trajectories. And we can also see that, so which is classical, that when NMC increase, we have the distance. So this is the L1 distance between the distribution of the initial system and the limit system, and we see that this distance decreases. So this is our theorem proves this. And another thing that we did not study yet is the speed of convergence, and we see that so this graph shows a bit like how it will converge when epsilon tends to 0. So how the distance between the two distributions will converge. So here it's for 5,000 trajectories of our process. So now I will illustrate the fact that we can, thanks to these computations, we can find parameters for which we have either convergence or divergence of our weights. So we'll take the spiking rate function to be a sigmoid like that. We'll consider the parameter, these parameters. And we observe that so with these parameters, when we take A plus to be equal to 0.2, we'll have our weights which will converge, so in distribution. And if we take A plus to be equal to 0.3, so if we just change a bit A plus, we'll observe divergence. So this can be obtained using our mathematical results. And a natural question when we have such results, we can wonder if the parameters involving convergence of weights in a 2-neuron network would also imply the convergence in a larger network. So we did this computation, and with 100 neurons, we observed that computing the average value of synaptic weights over time, we have convergence of our weight until a larger value of A plus, so until 0.5, and we have divergence approximately beginning at 0.6. So this may, so after this computation, it's interesting because if we can only find condition on the 2-neuron network, it costs really less than for bigger networks. So this is one of our perspective. is to show that the analysis of on 2 neurons is sufficient for the weight convergence analysis of more neurons. Also, what would be an interesting thing is to control the distance between the initial and the limit systems. So here we have our theorem tells us that when epsilon goes to 0, we have the convergence, and we studied the long time behavior on the limit model. So if we want to come back to the initial model, we have to control this distance between the limit model and the initial model using this, so yeah, in function of epsilon. So we could do, so we are trying to do a central limit theorem on our results to obtain such results. And then we would like to, there are many ways to extend our results and our models. So first we could put the times in the last spike S in the spiking rate, that which maybe, for instance, Gaëtan did this. Also, we could have a continuous membrane potential and try to find similar results, getting rid of the bounds on the spiking rate. And finally, what would be interesting is to investigate the memory capacity of this model. That's a thing that is commonly done in a model of a neural network with plasticity. So to do so, we have to define a learning protocol, and then we will have to define and study a learning to how the network learn a signal. So we could use the signal theory to do so. And finally, what is really interesting is to combine the mean field analysis and the slow fast analysis, because so we did a mean field analysis on this model, and in order to have the simplest possible model, we could hope that we can combine these two studies. So thank you for your attention, and if you have any questions, thank you.
Okay, thank you, Pascal, for this very interesting talk. We have time for questions. One of them is on your last point, the mean field analysis you mentioned. Is this numerical, or is this mathematical that you did?
So it's mathematical analysis. So we did. So we have some. So we had to assume the convergence of the empirical measure. And since we assumed this convergence, we were able to find the limit model. But okay. Yeah, so there is still this convergence that we have to prove to be to be complete. And yeah, we did some simulation to test this limit model, and it seems to work well.
Okay. And so if I followed you correctly, your membrane potential can take two values. Am I correct?
Yeah. It's
right. So do you expect difficulties in the adventure of going to continuous membrane potentials?
Yeah, the main difficulty would be to control this membrane potential to prevent it from going to infinity. But if you consider a model such as the one of de Massy and Fournier, in which you have a reset when you have a spike, you can expect to have the same results without a lot of effort, I think.
Thank you. I see a question by Priscilla Greenwood. She's asking convergence rate for many neurons, question mark.
Yes, Pascal, I liked your talk very much. And starting with 3 neurons, that's great. And then you went to 100 bravely, 100 immediately. And my question, and you still had convergence. And my question is, did you compare the rates of convergence? When you have so many neurons, does this slow down the system by some amount, or is it still converging quite nicely?
Yeah. So yeah, we can see. I didn't study it in detail, but we can see here that the average is still quite high after 300 seconds, and in the previous one, so yeah, I didn't. So in the previous one, we see that the weight, which is the bigger, stay far from 0 until let's say around 10,000 seconds. So yeah, I didn't go far enough to compare it. But yeah, because yeah, when you take so much neurons, and as I did computation, I take in memory all the values through time, so it's quite heavy with 100 neurons. That's why I didn't go further. So.
Okay.
Thank you.
Perhaps one last question by Sven Gödicke. In the limit, you consider the rates at which weight changes occur go to 0, but the amplitudes are finite? Question mark.
Sorry, what? Can you repeat the question? Sorry?
Yeah,
Sven,
I go online maybe.
Yes.
Yeah.
Online.
No, I was. Yeah, thank you very much. Very interesting. So I was just wondering. So how can I just like intuitively imagine the dynamics in the limit? So if I understand correctly, so because
I think I understood.
Yeah. So that the jumps of the weights are still finite, but they occur more and more rarely. Is this correct? Or
yeah, that's it. Yeah, usually you have the size of the jump will decrease with epsilon. So
yeah.
So you will have this fact that you need a lot of spike pair, and as you are looking on the fast time scale, you have a lot of spike pairs. So you observe on average something which is finite. Yeah. And in our case, it's thanks to this probability, which is slower and slower.
Yeah.
So smaller and smaller.
So
sorry. So if you look at on average, you will have a lot of spike pair, and you will see that there is one change of weight for all these spike pairs. Yeah. I don't know if it's it answers the question.
Yeah. Thank you very much. So I was just wondering how would you do this, and if you want to like simultaneously let the network go, or not simultaneously, but also consider like a mean field limit for the network size, then it would be difficult to keep this rarely, or
the mean field limit is we will consider we'll add in the spiking rate, we will add 1/n in the here.
Okay.
So yeah.
Okay.
And
sorry to interrupt you guys. You can
sorry.
continue the conversation offline, but thank you again, Pascal, for this very interesting talk. We have to move over to the next one by Rahul Gupta.
Thank you.
Can you start sharing?
I stopped?
Yes, stop. And
okay, it's okay. Thank you, Olivier.
Okay, so right, so this is from the Keno Donnell lab at the University of Bristol, and the talk is about the dynamical principles of synaptic receptor sharing across spines in a dendritic branch. The floor is yours.
Can you hear me?
Yes.
Yeah, thank you so much, Olivier. So what I'm looking for are what are the factors and how do they control the count of the synaptic receptors amongst the dendritic spines. So if you take a CA3 pyramidal neuron and zoom into a short stretch of its dendrite, you will see these granular projections, protrusions along the main shaft. And if you further zoom in, you can see the individual spines. Now, a single spine has a spine head and a spine neck, and the top section of the spine head's membrane, there is a dense raft of specialized proteins called postsynaptic density. The AMPA receptors are distributed all over the membrane, but they are present in this small region of postsynaptic density at a very high density. And this is due to the retarded diffusion of the AMPA receptors in the PST due to local macromolecular crowding, as well as the binding of the receptors with the PST95 slot proteins. Now, the importance of the count of these AMPA receptors in the PST is due to the fact that these receptors are closely juxtaposed to the active zone of presynaptic active zone of glutamate release, and thus predominantly shape the amplitude and kinetics of the excitatory postsynaptic potential. So the freely diffusing as well as the PST95 bound receptors in the postsynaptic density are together called by a spatial name, synaptic AMPA receptors, and the other receptors are considered extrasynaptic. Now, what are the factors which control the count of the synaptic AMPA receptors at a single spine and the distribution amongst dendritic spines have interested the neuroscience community for a long time. And a lot of studies has actually focused on single spine. Only very few studies, some in recent years, have actually tried to look at how the factors shape the distribution of receptors amongst the spines. Now, of course, there is a well-recognized immense molecular machinery for AMPA receptor trafficking, which shapes the unique distribution profile of the receptors on the dendritic arbor of the neuron. However, it doesn't work in isolation. One of the most prominent input is the presynaptic activity. However, the firing of the postsynaptic neuron also provides input to the molecular machinery through, for example, backpropagating action potential, as well as the local electrophysiological activities in the dendritic branches also provides input to the molecular machinery through, for instance, the dendritic spikes. So a straightforward approach would be to first study the machinery and then study the response of the machinery to the input. So the entire molecular machinery for AMPA receptor trafficking can, for easiness, can be divided into three principal mechanisms. One is the simple membrane receptor diffusion, where the receptors exhibit 2D Brownian diffusion in the phospholipid membrane with a diffusion coefficient of 0.1 micron meter squared per second. But with a 1/10th reduced diffusion coefficient in PST due to local macromolecular crowding, as I mentioned before. And this receptor diffusion causes exchange of receptors between the somatic membrane and the dendrite, parent dendrites and the daughter branches, and eventually between the spine head compartments. The other very important mechanism of AMPA receptor trafficking is the exocytosis and endocytosis, where there is a constant exchange of receptors between the membrane and the intracellular receptor pool. And this happens in the spine head as well as the dendritic shaft. And the intracellular receptor pool is constantly affected by the local AMPA receptor synthesis as well as degradation, and the anterograde and retrograde vesicular transport of AMPA receptors. So this leads to the third important mechanism, which is the motor protein assisted active vesicular transporter proteins, AMPA receptor proteins, where the large cargo vesicles carrying these AMPA receptors move in through anterograde transport into the daughter branch, and the receptors from the intracellular pool are transported back to the somatic compartment, loaded on these cargo vesicles, again walking on the microtubules through the parent dendrite. So if one tries to think about an integrated biophysically detailed model for understanding how these three mechanisms shape the count of synaptic AMPA receptors, it becomes very daunting. Now, the point is that each mechanism is regulated by thousands and thousands of proteins and signaling factors, and many of them are undergoing nonlinear computations. For instance, the simplest, even if I look at the receptor diffusion, then the receptor diffusion is very much affected by the size of the intracellular C-terminal domain of different AMPA receptor complexes, the phosphorylation status of the terminal domain, C-terminal domain, and the association of the AMPA receptors with tau proteins such as stargazing. So thinking about an integrated detailed model is indeed daunting, as well as also limited from the point of parameter estimations, because many of the details we still don't know from the experimental point of view. And if suppose someone even comes up with this model sometime, some years later, then this will of course prove to be extraordinarily computationally expensive, and it will be difficult to actually develop a mechanistic understanding. So this necessitates a coarse-grained rate mass model, and here I consider a compartment-based reaction diffusion approach, where what I do is that I take a daughter branch coming out from a parent dendrite, and I chop it like a carrot into small compartments, and each small dendritic compartment, the density of the receptors in the membrane of that compartment is shown as R subscript dent, and the intracellular receptor pool in that small compartment is clubbed together, and there is a crosstalk between the membrane and the intracellular receptor pool through endocytosis and exocytosis. Now, the intracellular receptor pools are also constantly enriched with new receptors through anterograde transport, as well as depleted by the retrograde transport. And the anterograde and retrograde transports are independent for each compartments. Now, some of the dendritic compartments also bear spines connected through the spine neck, where the density of the receptors in the neck are shown as R subscript neck, and the density of the receptors in the extrasynaptic membrane shown as R subscript ESN. Now, the spine head also contains an intracellular receptor pool defined as IR spine, which shows the similar mechanism of endocytosis, exocytosis, retrograde, and anterograde mechanisms. And the pool of the receptors in the postsynaptic density are divided into two pools, subpools. One are the free receptor pool and one the bound receptor pools, where there is an exchange between these two pools based on the density of the PST95 slot proteins, binding and unbinding parameters. So this coarse-grained model actually provides a very simple mechanical structure capturing the most essential interactions for trafficking and binding processes. Now, this model works on the system of mostly linear ODEs, where only nonlinear reaction terms are for the binding of the receptor with the PST95, and this is just because of the binding depends on the availability of the free PST95 slot proteins. And this model actually works on the list of on these parameters, where there are geometric parameters which define the shape and size of the compartments. Then we define a boundary condition at the initial starting point of the daughter branch, where we are considering a steady concentration boundary condition. And then we have hopping factors, which define the exchange of the receptors between the different compartments through AMPA receptor diffusion in the membrane. And then we have intracellular receptor trafficking parameters, where we have anterograde and retrograde parameters for all the spine heads and dendritic compartments. We also have membrane trafficking membrane receptor trafficking parameters, where we have parameters for the exocytosis and endocytosis, and for all the spine head and dendritic compartments. And then we have PST95 density, binding, and unbinding parameters for every PST compartment in the spine. Now, if we consider the dendritic branch as a system, then the anterograde parameters and the boundary condition actually causes influx of receptors into the system, and the retrograde ones actually deplete the system for the receptors. Now, when we try to actually compute the steady state solutions using the numerical integration of this large ordinary differential equation system, then the problem which happened is that as you will see afterwards, the parameters are varying in their orders of magnitudes from 10 to the power minus 4 to 1 per second. And that leads to a strictly stiff ordinary differential equation, and reaching the steady state solution even were taking minutes of computations. But we actually developed the recurrent structure, which is a novel structure for this chain of compartments, where we can quickly, within seconds, compute the steady state solutions, where if we just need the steady concentration of the receptors at the boundary condition and the parameters, and we can backtrace within seconds the density of the receptors in each compartment. And this recurrent structure actually helped us to examine thousands and thousands of parameters to see how the parameter combinations are actually affecting the distribution of the synaptic AMPA receptors in the spines. Now, here I have given the explicit expressions of the steady state solutions, although I won't go into the details, but this is there in the slides, which is available in the Dropbox. So if one is interested in understanding how is this recurrent structure working, then one can have a look at it. Now, here I am now very soon going to dive into the result section. So the geometrical and reaction parameters, which I'm using here for the results, I would like to mention, where the dendritic shaft, the daughter dendritic branch is of a diameter of 1 micron meter, and the length is 100 micron meter. And the small compartments which I have created in that is of the length of 0.1 micron meter. And the neck of all the spines are identical, of diameter 0.4 micron meter and length 0.5 micron meter. Area of the ESM and the PST is also identical for all the spines, and area of the ESM is 1 micron meter and PST is 1 membrane area, and the interspine distance is 1 micron meter. The diffusion coefficient I have already mentioned, and the steady density of the AMPA receptors at the boundary condition, which I'm considering, is 10 per unit micrometer square area. The PST95 binding and unbinding rates are 10 to the power minus 4 per second and 10 to the power minus 5 per second. And the PST95 density available for the AMPA receptor binding in the PST is 150 unit per unit micrometer squared. So in the area of 0.1 micrometer squared of the PST, we have 15 PST95 slot proteins to bind. And these parameters are more or less inspired from the mean empirical estimates available in the experimental literature. So now in part A, what I'm going to show you is how the parameters anterograde, retrograde, endocytosis, and exocytosis, the 4 crucial parameters of AMPA receptor trafficking, may affect the distribution of synaptic AMPA receptors across spines. So we begin with a situation of free diffusion in the PST. So we are, for the time being, ignoring that there is a hinder diffusion of AMPA receptors in the PST, so that when we want that when we apply that, we can see what change does it make. So we start with the free diffusion condition, where we take the dendritic branch and we look at the count of the total count of the synaptic AMPA receptors and the PST95 bound count of the AMPA receptors in the first spine. So what we do is that we take 5 different orders of magnitudes of the endocytosis and exocytosis parameters and combine them into this form, and then we try it for 5 different orders of the anterograde and retrograde parameters. So the orders are 10 to the power minus 4 per second, 10 to the power minus 3, 10 to the power minus 2, 10 to the power minus 1, and 0. So I have shown here in terms of the log of the values. Now, the heat map is actually showing you the count of the total synaptic receptors in the PST, and here the PST95 bound count. And what we observe is that most of the parameter combinations are actually leading to the saturation of the PST95 slot proteins, and there are only few of them which are actually leading to unsaturated or partially saturated PST95 binding. As well as we also see in the total count that the total count is close to the, for most of the parameters, close to the count of the PST95 bound count, which is maximum up to 15. So as many available slot proteins are there, and the range in the total count in this color zone is 1 to 20. So the thing is that the free receptor counts are very less in comparison to the bound count, and most of the composition of the total count is being constituted by the PST95 bound count. Now, I would like to show you the profile of variation of how the total PST receptor count and the bound PST receptor count actually varies for this combination of the parameters. So we chose this way because this is the best way we could see to actually express the result for 4 parameters. Otherwise, it would have been very difficult to capture this picture for a 4-dimensional profile. So we see that for these parameter combinations, we observe that there is a decrease in the profile, but also for another parameter combinations, we have an increase in the profile. So since the in this parameter combination, the PST95 bound count is already saturated, so you see a very fractional change. But the real change which is happening in the total PST receptor count is because of the change in the free receptors diffusing in the AMPA receptors. Now, to capture such profiles for almost all the parameter combinations, we devised a technique that we compute the percentage change in the total count of the receptors in the last spine with respect to the first spine. Now, the percentage change actually helps to inform on the how fast. So if we would have taken the absolute change, we could not have understood how fast the decrease or increase is happening, because that depends on what is the baseline value on the first spine, but taking the percentage change actually removes that effect and helps us to understand how fast it is changing. And we observed that for some combination, the total count is not varying much. For some combinations, it's varying more than 100%. And for some combinations, we observed that the total count decreases by 100%, like goes to 0 value to the last spine. And for the PST95 bound, we observe bound count, we also observe the same, but we only see the decrease because one can't increase the count of the PST95 bound count more than the available slot proteins. So that restricts the distribution of the bound count across the dendritic branch. Now, when we apply the reduced diffusion in the PST, the trapping PST, we observed a very different scenario where the free counts also increased significantly in the spine, as well as more and more parameter combinations. If you see many of the parameter combinations which were leading to partially saturated PST95 bound count have now become fully saturated, and the variation in the PST95 bound count has also gone low for many of the more parameter combinations. Now, because of the change in the free receptor, the change in the percentage change across the dendritic branch in the total count looks a bit more complex, but we did it for many more combinations. Here I'm showing you only for 2 situations. And what we conclude is that the ratio of the anterograde supply to the retrograde supply actually sets the baseline of the receptor count in the first spine, and then also decides what will be the direction of the change and how much will be the change, how fast will be the change. So higher is the anterograde supply to the retrograde, higher is the first spine count, and the profile will increase with faster rate. Now, given the baseline set by the anterograde and retrograde, we observed that the relative strength of exocytosis to induce cytosis performs a fine tuning where higher is the ratio, higher is the spine count, and faster is the change in the profile set by the anterograde and retrograde combinations. Now, as I showed that the PST95 for most of the combinations are actually crucially shaping the receptor count, and most parameter combinations exhibit fully saturated bound receptor pool. And trapping PSD significantly increases the free receptor pool and reduces the variation in the bound receptor pool down the branch. We have also studied one effect that is empirically observed in the dendrites, that the parameters of the trafficking mechanisms in the dendrites have been observed to be an order less than the parameters or in the spine head. And when we tried it, we observed that it even further reduces the variation in synaptic receptor count. So this factor actually stabilizes the count of the synaptic receptors across 100 micron-meter dendritic branch. But we don't know if we make it like 200 micron-meter dendritic branch, how will it be affected? But that's the estimate we have taken up. And then it also explains these profiles also explain why the spine sizes in some dendritic branches, if you see, as you go distally, then the spine sizes increases, and in some branches it decreases, and that has always been mysterious why. But using these homogeneous parameter combinations, we have observed that maybe this is a homeostatic mechanism to accommodate, to compensate for the loss or gain of the receptors across the dendritic branch given different combinations. So with this, I will now jump on to the part B, where I would like to show you how the heterosynaptic effects during LTP and LTD may depend on these parameter combinations. So here, when I am applying LTD in my model.
Gupta, let me remind you that you have about 5 more minutes.
Yeah, I'm looking at the time.
Thank you.
So there's a version 1 where I'm just keeping the spine head size unaffected, but version 2 where I am shrinking it, and in the LTP where I keep the spine head unaffected for version 1, only increasing the PST95 density, but in version 2 I am enlarging the spine head and decreasing and keeping the PST95 density unaffected. Now what we observe that in the trapping PSD situation, the LTD version 2 has more strong heterosynaptic effects than LTD version 1. In the case of bound counts, we don't see much heterosynaptic effects because the PST95 bound receptors are already saturated to their maximum binding slot proteins. And similarly, we observe in the case of trapping PST that the LTP version 2, where the spine head enlarges, that in that case we have a stronger heterosynaptic effect. Now, if we remove the trapping PSD situation, we observe that the well-performing LTD version 2, where the spine head was shrunk, and the LTP version 2, where the spine head was enlarged, have now lost their performance, and the heterosynaptic effects have gone down. So with this, I would like to conclude the part 2, where we observe that the set of trafficking parameter combination, which leads to similar synaptic receptor distribution across the branch, does not necessarily mean that they will also exhibit similar strength of heterosynaptic plasticity. And we observe that empirically observed the shrinkage of a spine head during LTD and enlargement during LTP are potentially more effective in heterosynaptic effect than near changes in the PST95 density. And the heterosynaptic effects also are observed to happen with a delay So at the point when I am doing perturbation in the middlemost spine on the dendritic branch, the neighboring spines are getting this perturbation effect with a certain delay, and the significant changes in the free receptor count is only staying for a limited window. So maybe this limited window has an implication that might be used for establishing a correlation between the LTP and LTD events amongst neighboring spines. And the length of the dendritic branch has been seen to affect a lot the amplitude and time kinetics of the heterosynaptic effects. So with this, I would like to conclude this presentation, and I would like to thank the Leverhulme Trust for the independent research funding that they are providing, and the University of Bristol for the excellent research facility and workspace. I would also like to thank my research supervisor, Kian O'Donnell, for time-to-time suggestions, and my colleagues, members of the Computational Neuroscience Unit, and my lab members for their critical suggestions. So thank you so much for the patient listening.
Oh, thank you very much. You kept your time, Raoul. We have time maybe for a couple questions, maybe one or two, if there are questions on the chat. It's a bit unclear to me what kind of mathematical techniques you're using. I mean, I saw a bunch of ordinary differential equations. Any other techniques that you are using?
Yeah, so for like in this case, like here, the ODEs, so my model is mainly based on ODEs. We are not using partial differential equations. Most of the part, it is linear ODEs, but we are introducing more and more non-linearities in the ODE section. And then we are also involving stochastic differential equation formalism, where we are converting those ODEs into stochastic formalism. And we are also looking at the statistical properties of the receptor distribution.
Okay, so in the SDE case, what kind of noise are you working with? Standard Brownians?
So I'm right now trying to use, we are working on the additive noise as well as multiplicative noise. And in the noise section, we are right now trying to use the Gaussian noise process. So.
And they are motivated by experimental measurements?
Yes.
Okay. All right, I don't see any more questions on the chat, so I think, well, thank you again, Raoul. Apologies for this.
Thanks, Olivier, thanks.
And thank you for this very interesting talk, and thank you to all speakers who attended this session. We are now going to another session. I see Willem Stana's face on a different topic, which is the future of ICMNS.
Yeah, thank you. Thank you very much, Olivier, for organizing the mini symposium. Thank you for all the speakers and the mini symposium. Just before you leave, I just want to remind you that we reconvene tomorrow already at 2 p.m. and not with a panel or plenary talk, but directly with the mini symposia. So directly choose the channel or the mini symposium that you would like to see. And this is then followed by the micro talks. And today there is a special panel discussion on the future of the ICMS, and we start in 10 minutes past the hour, so at 6:10. See you then.
It's not right now. Okay, it's in 10 minutes from now.
Yeah,
okay.
We have a small break for this.
Small break. Oh, thank you. Thank you. Okay, talk to you later in 10 minutes.
Yeah, see you.
See you.
Daniela?
Yes.
So are you there? So should we
Yes.
stop the YouTube channel or? I mean, I'm
Whatever
open
you
to it.
prefer.
Well, I don't know. So
What do we want to do?
I think we should stop it.
Yeah. Let me stop it here. In fact, let me also stop sharing.